Hallo iedereen. Hello everyone. We're going to start in 2 minutes. This session will be streamed online, so we will start in time at 12 p.m. Sharp. I'm your host today. I'm Maarten de Hussem. I'm a poultry veterinarian and consultant, and I've been asked to host you this time in this wonderful room here in Amsterdam. We had a similar session a few years ago, and then I was one of the speakers, and I can tell you it's a very nice room because everyone's very close together. There's a very good acoustic in the room, and I'm sure that we'll have a great time this afternoon. I'm looking a bit at the time for the streaming, but maybe you can indicate if the streaming has started. Then I can know when we can start our session. All right, we're online, so be quiet, please. So today we have a program with basically two different sessions. The first session will be focusing more from the animal health angle, and both Evy Goossens and Tim Goossens. So you see, it's a very common name in Flanders to have the name Goossens. So we have the two Goossens that will kick off and look at gut health from an animal health angle. That will take about 1 hour and 15 minutes, and then we'll have a lunch break. And after the lunch break, we change the angle a little bit. We go from the nutritional angle, and then we will have two more speakers, Francesc Molist from Schothorst Feed Research and Christy de Wildt, which will talk about lysolecithins, and she's an internal FRAmelco expert. Okay. First of all, you see a lot of FRAmelco here. I'm not FRAmelco, I'm independent, but I'm your host, and it has been a while since we have been able to meet each other in live, and it's great to be here this week with the VIV. But you might have noticed that that things, in the meantime, business-wise, have been changing. And I was just asking Paul if he could just elaborate a little bit on what happened. Last time when we were here, there was like FRAmelco everywhere, and now we see a small other addition to that name. And I was just wondering if you could just share some ideas and thoughts about what happened.
Correct. Thank you, Maarten. So, yeah, we are in a dynamic time. So, as you all have known, FRAmelco was taken over by Adisseo. And I just want to go a little bit back, actually, where the company started. And the company started as under the name of Franklin in 1977. So we are already a long time already active in this business. And later on, in 2007, we started with the current range of products we have now, the FRA range of products. Shortly after that, in 2008, we were one of the pioneers on glycerides. And glycerides, you have seen the last years are really catching up in the market, and also the results has been seen. You will hear a lot about that. And we started firstly with the buy-in, the glycerides, but a few years later we also started production in Spain for the glycerides. Later on, in 2014, we also have started with the lysolecithins. That are the hydrolyzed lecithins, and also shortly after that we also start to produce them in Spain. Only what I want to tell additional, that we also in 2019 we expanded also with a factory in Thailand for the mixing, and as already mentioned, finally we were taken over by Adisseo in 2020, and we expect a lot in the next years to grow further in this combination. So I wish you a very nice meeting today, and I hope here in the room, and also if you are online, I hope you will enjoy all the information coming up. And now I give the speaker of the microphone back to Maarten.
Thank you, Paul. So let's move then to the more formal program. We have the first speaker that I would like to invite on stage as well. She's all wired just before we started. Dr. Evy Goossens is leading a subgroup of the Van Immerzeel lab in Ghent University. She has 5 PhD students and 1 lab technician reporting to her. The group mainly focuses on host microbiota interactions and how this influences both the intestinal and extra-intestinal processes. She's more specifically focusing on a relationship between microbiota and host during intestinal colonization, and their specialization is really lesion development in infections with Clostridium perfringens, not only poultry, that's where I think it started basically, and that's where the group is best known, but they also look into Clostridium perfringens lesions in other animal species, broadening the spectrum, in fact. A second tightly rated research topic is very close to what we will discuss today also, because they are looking at the effect of biological molecules, for instance, chemicals and bacterial enzymes or metabolites on both the gut microbiota, but also on the host of that gut microbiota. And Evy will today talk about, let's say, an update on necrotic enteritis and Clostridium perfringens in poultry production. Evy, the floor is yours.
Thank you. Okay. Thank you for the introduction. I will start with skipping some slides because they asked me to shorten it a bit. Sorry. So we'll start with Clostridium perfringens. Necrotic enteritis is caused by Clostridium perfringens, but Clostridium also causes a lot of other diseases. And Clostridium perfringens is an anaerobic gram-positive spore-forming bacterium that is auxotrophic for 13 amino acids. So this means that it has to acquire these amino acids from its environment. Perfringens is widely distributed in the environment, such as the soil and water sewage, and it can also be found as a normal member of the intestinal microbiome. But Perfringens is also a really important pathogen, and that's a little bit strange because it's auxotroph for these amino acids, so it has to acquire it from the environment and it can't produce it themselves. So to cause disease, Perfringens destroys its tissue, the host tissue around it, and as such, it causes disease. So there are a lot of bacteria that use tissue from the animals, but mainly when they are dead. Perfringens does this already in live animals. So that's what makes it so pathogenic. So depending on which Perfringen strain you have, different toxins are produced. And these toxins are then responsible for disease and are used to divide Perfringens in different toxinotypes. So depending on the strain and the toxins that Perfringens produces, it causes disease in different animal species. You have certain toxins that are commonly present and are universal, like the alpha toxin, which is needed to cause gas gangrene in various species. And then you have other toxins that are species-specific, for example, the NetB toxin, that is important for necrotic enteritis in broilers. So necrotic enteritis. Necrotic enteritis can be seen as 2 different forms. So on the right side, you see the clinical necrotic enteritis, where the animals drop dead. And when you open the small intestine, you see this necrotic tissue, like a complete necrotic layer covering the intestine. But the more common form is the subclinical necrotic enteritis, where you don't really see anything to the birds, but when you open the small intestine, you see these necrotic lesions. And this will cause a huge impact on the feed efficiency, the nutrient uptake, and the growth of the birds. So to study necrotic enteritis, we use an experimental animal model, and for this we have the chickens and a pathogenic strain. But if we just give this bacteria to the birds, they don't develop necrotic enteritis. For this we need a lot of predisposing factors. And these can be either immunosuppression, coccidiosis, which is a really important one. This causes leakage. Yeah, it causes intestinal damage, and it causes leakage of plasma proteins in the intestinal lumen, which is then used by the Perfringens to grow. We also use a high stocking density, which causes stress, and feed with fish meal, which is again a protein source, which is important for Perfringens because it can't produce the amino acids by themselves. And then, of course, we need the pathogenic strain. But even if we use all those predisposing factors, not all birds will develop necrotic enteritis. So what we see is a high variation in disease severity in our trials. Some animals don't develop necrotic enteritis, and others develop some lesions, and some others develop a complete necrotic intestine and almost drop dead. So we wondered what is actually the cause of this difference in disease severity. So we tagged the birds before the experiment, and we followed them throughout the experiment. So we weighed the animals before the coccidiosis challenge, before the Perfringens challenge, and in the end of the experiment. And then linked this to the severity of the lesions that were present. So what you see here is the weight of the birds before we challenged them with Perfringens. And this is the outcome of the disease. So the birds that did not develop lesions have a quite low weight before challenge. These are all birds that developed lesions, the mild ones and the severe. And what you can see is that the most heavy birds before the challenge develop the most severe lesions. And when you look after challenge to the weight, you can see that this weight benefit that these animals had is completely reversed. So yeah, it looks like the most well-growing and the most protein-using animals are predisposed to get the most severe necrotic enteritis. So using this model, a lot of different studies are done. One of them is to find out which Perfringen strains are able to cause necrotic enteritis. So what you see here is the percentage of animals that have lesions. When you inoculate the birds with just a bacterial culture medium without the Perfringens, and then you see none of them get necrotic enteritis. If you use only the paracox, so the predisposing coccidiosis infection, none of them got necrotic lesions. Also, with these three Perfringen strains, no lesions were observed. And with these three, we could see a variation in disease severity. And the main difference between those strains is that these produce an NB toxin and these don't. So this indicates that NB is really essential to cause disease. But to be absolutely sure that NB is a causative toxin, you need to use knockout mutants. So the group of Keyburn, they used a pathogenic strain. They made a knockout in the strain, so it couldn't produce the NB toxin anymore. And then this strain was used to complement it again. So there you have the knockout strain that again can produce the NB toxin. So these three strains are essentially completely the same. They produce everything the same except the NB toxin. So if NB is really essential to cause disease, you would expect disease here, nothing here, and then this strain should again cause necrotic enteritis. And this is indeed what you see. So we have lesions with the wild type strain. If you remove the NB, there's no lesions, and then if you put it back in, they can cause disease again. So we can say that NB is important for necrotic enteritis. So as a summary, Perfringens is really limited in its amino acid biosynthesis, and this is really important. It needs proteins from its environment to grow. And to do this, it produces toxins to break down the host tissue for its own benefit. And specifically for necrotic enteritis in broilers, it uses NADB. But you don't only need the Perfringens, you also need predisposing factors. And the most important one is the protein, and which can be the coccidiosis, which causes leakage of plasma proteins in the lumen, or a high protein feed. And there's also a high variability in disease severity, and heavier birds seem to be more susceptible to disease. So in 2008, the NADB toxin was identified as the causal toxin for necrotic enteritis. And the researchers thought, now we have solved the problem, we will focus on the NADB toxin. And there was really a lot of research focused on this toxin. And people tried to make diagnostic tests and vaccines based on the toxin. But still, the problem is not solved. So this can also be seen in the toxinotyping scheme. So what you see here is Perfringens can be classified in different toxinotypes. And this is done based on the toxins it produces. Well, a selection of those toxins. You can see the alpha toxin, which is produced by all strains. And then you have the other toxins, like beta, epsilon, and iota toxin, which are used to classify the Perfringens. For example, the type C strains produce alpha toxin and beta toxin. And this part here was a classical toxinotyping scheme before 2018. But now, because of this recap feeling that NADB is really important for the disease that we are studying in chickens, these were added to the toxinotyping scheme. And we also have type G strains now, which produce alpha toxin and NADB, which is important to cause disease in chickens. But the question is NADB really the holy grail, and is it really the only thing we should be focusing on? And indeed, we see NADB producing strains that cause necrotic enteritis around the world. But there seem to be more than just this. In the US, Canada, and China, you have research groups that report that they are unable to isolate NADB-positive strains from necrotic enteritis cases in the field. And they seem to be able to induce necrotic enteritis again in the animals using their NADB-negative strains. So we were wondering, are they looking to the same disease, or are they describing something as necrotic enteritis that is maybe something else? So this is what we see in Europe, and which is caused by NADB. So you have the small intestine that is completely filled with gas and distended, and when you open it, you see the necrosis. And on histology, you see gram-positive rods, so Perfringens, at the necrotic tissue. And there's mostly a co-infection or a preceding infection with Emeria. So coccidiosis is really important. But then you have this, a hemorrhagic form of necrotic enteritis. So you see that the intestine is filled with blood, and the mesentery, the vessels are congested and filled with blood. And when you open the intestine, you see again the necrosis, but also a lot of blood. So the main difference here is the blood. And this is what is described in the US and Canada as being necrotic enteritis. And the most important thing, this is NADB. The hemorrhagic form, it's not NADB. And we don't know what is actually causing this disease. So both diseases are seen in conventional broilers. So we have the standard non-hemorrhagic form of necrotic enteritis that we are all studying that is caused by NADB. Then you have the hemorrhagic form that is caused by an unknown toxin. So we propose to rename this disease as necrohemorrhagic enteritis to make clear that these are two different diseases and that we are not talking about the same thing. But then there's something more than just conventional broilers. More recently, people are more interested in alternative breeds, such as slow-growing birds or organic broilers. And these birds, they are the result of highly heavy genetic selections why you get different breeds. So you don't only get difference in overall physiology, which is quite obvious, but also the metabolic processes and immunity management and everything of these birds is different. So the question is there one C. perfringens type, like we now have the type G, that is capable of causing disease in all these breeds, or do we have different toxins, different types that are needed to cause disease? And another question is, as they are so different, are these breeds all equally sensitive to necrotic enteritis? So to answer this question, we had a look at the Belgian prevalence data from 2019. And what we saw was quite striking. We didn't see any necrotic enteritis in the conventional broilers. But then when you look at the alternative broilers, the slow-growing birds, we had 9 to 10% prevalence of necrotic enteritis. And then you see the organic broilers, which is quite impressive. There we have up to 40% of necrotic enteritis. And here this is the main disease in organic broilers. When we then look at the age at which the birds are at risk for necrotic enteritis, we see a peak in the slow growers between 2 to 3 weeks, and in the organic broilers there are 2 peaks, one at 2 weeks and one at 4 weeks. So the interesting question is, of course, what is causing the difference in this disease susceptibility? So because we know that coccidiosis is an important predisposing factor, we had a look at the coccidiosis management between those different types of broilers. So what you see is that at that time in Belgium, we used mainly vaccination for the slow growers and the organic broilers. While in the conventional broilers, mainly in-feed coccidiostats are used. So this is interesting because we know that ionophoric coccidiostats, they target the Emeria, but they are also antibacterial for Clostridium perfringens. So it might be that these coccidiostats are both targeting the predisposing infection and the perfringens itself. But this, probably other things can play a role as well, but it looks like a possible explanation. But then there's still a big difference between those 2 types of broilers. So the question is, what is causing this difference in sensitivity, and why are these organic broilers so heavily affected? And that's something that we still don't know. So to summarize, current research is completely focused on conventional broilers, and then mainly on the non-hemorrhagic form of necrotic enteritis that is caused by NET-B. But there's more. In other parts of the world, there seems to be a hemorrhagic form of necrotic enteritis, which is not caused by NET-B. It might be a novel toxin, but we don't know what is causing this. And there's even more. We have a really high prevalence of necrotic enteritis in organic broilers, and also in slow-growing birds, a high prevalence is seen. We don't know why we have these high prevalence, and we don't know which toxins are important here. So more research is needed on these alternative forms of necrotic enteritis. So when we were doing research on necrotic enteritis, we were, like everyone, focused on broilers. And we wanted to develop a test, an assay to detect necrotic enteritis in the feces. So we asked to the veterinarians if we could get samples from necrotic enteritis affected flocks. But to our surprise, we didn't get any samples from broilers. We got a lot of samples from layer pullets. So that's why we looked further into the layer pullets. So we first looked in the literature, and there was almost nothing to find. We have 2 quite old papers that describe necrotic enteritis in the layer pullets, which is the same as we see, so before they start laying eggs. And the same as with broilers, you see there's a presence of Emeria and coccidiosis, which is associated with the necrotic enteritis. But although there's nothing in the literature, we seem to have a high prevalence in Belgium. So for this, we looked at the veterinary intervention records from the two largest poultry veterinary practices in Belgium, and we looked at a period of 5 years. In this period, there were 23 pullet farms in the data set, and together these were 295 different flocks. And when you look at the main reason for which the veterinarian is called in the pullet farm, you can see that coccidiosis and necrotic enteritis are the top reasons why the veterinarian has to go. If you then look at the macroscopic presentation of the disease, you see something that is really similar to the image we see in the broilers. So you have the small intestine that is distended and filled with gas, and when you open it, you see this necrotic tissue, and in really severe forms, you see these necrotic plugs that are completely detached from the underlaying surface. On histology, you can see a lot of coccidia on the necrotic tissue, and Perfringens-like bacteria. And here you see the muscle tissue and then viable crypts. And on top here, you see the necrotic tissue. And what you see here is also really similar to what we see in necrotic enteritis and broilers. You see this infiltration of inflammatory cells that are like separating the necrotic tissue from the underlying viable crypts. What you see here are the viable crypts, and the necrotic tissue that is completely detached. So that's actually the image here. So to look further at the incidence, we can say that in our case we saw 12% of all layer pullet flocks necrotic enteritis in Belgium, and the mean age was 10 weeks where they got necrotic enteritis. Because coccidiosis is also really important as a predisposing factor, we also looked at coccidiosis, and in this data set, around 50% of the animals, the flocks, were affected with coccidiosis, and this was a little bit earlier. And what is even more interesting is that we saw a clear link between the necrotic enteritis and the coccidiosis, and farms that have coccidiosis are 2 times more likely to develop necrotic enteritis. But because of, we saw a breed difference in the broilers, we also wanted to look at the breed here. The actual breed of the birds was not present in the data set, but we know the color of the birds. So we have brown-feathered and white-feathered birds. And what you see here is the total number of flocks with brown-feathered birds, and then the amount that got necrotic enteritis or coccidiosis, and the same for the white-feathered birds. So what you clearly see is that there's a high fraction of the flocks in the white-feathered birds that got diseased. So there's a higher prevalence in white-feathered birds. So, of course, the interesting question is there a link with the net B toxin? So are we talking about the same as in the broilers? So to study this, we collected samples from diseased birds. So we took a swab from animals from 10 different necrotic enteritis outbreaks, and we took a swab from the lesion. And to have a comparison of a healthy Clostridium perfringens population, because it's present in the normal microbiome, we also swabbed 8 different flocks where no history of necrotic enteritis was present, and here we took a cloacal swab. And the interesting thing is that we couldn't isolate Perfringens from all birds here, but only from half of the flocks Perfringens could be isolated. So this indicates that Perfringens in a healthy flock is not at really high levels. We also have samples from healthy birds within a diseased flock, but these are not so reliable as a healthy control population because the same strains can be present in these birds. So to see what is special in this isolates from diseased birds, we did a whole genome sequencing on a selection of strains from the necrotic lesions and from the healthy birds. And then we compared the whole genome of these isolates And what is interesting is that we didn't find NetB toxin in any of the isolates. We screened also a larger collection of strains, and also there no NetB was found. What is even more important is that we didn't find any unique toxin genes or novel toxin genes. And we know that, for Perfringens, it is commonly accepted that it causes disease by producing a toxin, and for each species you need a specific toxin, but in this case we didn't find anything. So you could say that maybe we isolated the wrong strains, but we did find a unique region of 6 genes, which is uniquely present in the pathogenic strains. And because of this, we call it the padlock region. So the interesting thing about this padlock region is when we screen our complete collection of strains, we see this region present in every outbreak of necrotic enteritis in layer pullets, but it is not present in any other published C. perfringens sequence. So it's not present in any strain that is isolated from the broilers. It's not present in the strains that cause enteric diseases in dogs or horses, or the strains that cause gas gangrene or food poisoning in humans. So it's something really specific for the layer pullets. And when you look further at the clustering of the strains based on the phylogeny, you see that these padlock-positive strains form a distinct phylogenetic cluster as compared to the other strains. So to summarize, in layer pullets, we have a high prevalence of necrotic enteritis. We see non-hemorrhagic lesions and a predisposition of, due to coccidiosis, which is similar to the broilers. It is not NetB that is causing disease, but there is a unique genomic padlock region that is associated with necrotic enteritis. We don't know what the function is of this region. We also don't know why we don't find the NetB positive strains in the pullets. I can say we do find NetB positive strains in adult layers, but never before they start laying eggs. And we also don't know why we don't find these padlock positive strains in the broilers, although they are also young animals. So there seems to be a difference in colonization. So to focus further on this initial colonization, I want to go back to the inside of the intestine. So what you see here is the intestinal lumen, the intestinal villi, and here you have the inside of the intestine. This is based on research, so this is an example of what is happening for NetB positive strains in broilers. So to cause disease, these are the Perfringens strains. They produce the NetB toxin, but the NetB toxin has to pass through the mucus layer, the epithelial cells, and then through the epithelial layer to the place where it causes disease. And we know it's not one of these structures. The toxin targets something that is deeper in the tissue. So that's what you also see on histology. You see Perfringens attached to the epithelial cells. It is multiplying and then causing the disease. So to focus on the first part that it has to overcome is the mucus. So Perfringens, the mucus is mainly composed of mucins. And this is something really complex. So the most important thing that you should know is that this is a protein backbone. And on this protein backbone, you have a high diversity of glycans. And there's a different composition in these glycans between different species. And specifically for chickens, there's a high level of sialic acid there. And Perfringens produces a high variety of mucus-degrading enzymes. So each bar represents a different enzyme. And the thing I want to show here is these are crystal structures of different enzymes. But the thing you should know is that you need a different enzyme to cut every branch. So that's why Perfringens has so many. And it's the same as with the toxins. One Perfringens strain will not produce all these enzymes. It produces a selection of it. And by this, it can tailor its needs to the specific mucus composition that it has, that the animal has. So you get a species-specific glycan degradation. So to make it a little bit easier, the mucus is composed of a protein backbone. Then you have the glycan trees, and on top of the glycan trees, especially for chickens, you have a lot of sialic acid. So when Perfringens needs to dismantle the mucus layer, it has to start with the sialic acid, because this is covering everything. So the first thing it has to do is remove the sialic acid. And for this, Perfringens has different sialidase enzymes, and the most important one is the NAN-I enzyme. And this NAN-I enzyme will cut off the free sialic acid, which is then used as a nutrient for bacterial growth, but it will also induce the toxin expression, so you get a lot of toxins that are later on needed to cause disease. And then what is left is your glycan structures that are now accessible for the other enzymes to break it down. And also on the epithelial cells, you have these glycans. And here, if the sialic acid is removed, the Perfringens strains can adhere to binding sites that are unmasked due to the removal of the sialic acid. So what we have now is Perfringens attached to the epithelium, as we saw on the histology. There's already toxins produced, and the glycan trees are accessible for further breakdown. So the next step is the breakdown of the glycan side chains. This can be done by a lot of enzymes, as we saw before. So we have the hexosaminidases, galactosidases, but what is interesting for the NADB-producing strains, they have chitinases, which is only seen in the NADB-positive strains. So yeah, chitinases. This is a bit weird in a chicken because chitin is the second most abundant polymer on Earth after cellulose, and it's found in the outer skeleton of insects and shrimps and internal structures of invertebrates. And it's actually just a linear polymer of glucosamine. So you have this monomer repeated a lot, and then you get a chitin. But the chicken is not an invertebrate and it's not an insect, so it's a bit weird what the chitinase is doing in a chicken. So when we look at chitinases in other pathogens, we see that Legionella pneumophilia produces chitinases, and these are important for bacterial infection in the lungs. Then you also have Listeria monocytogenes, which produces chitinases that are important for colonization of the mouse intestine. And then we are focusing on the mucins. And we know that mucins, and on top of the vertebrate cells, there are these glycans where you have those glucosamine structures present in there. So probably the chitinases from our Perfringens strains are important to break down the mucus. So to investigate this, we did a binding assay. So we have here our two chitinases from Perfringens, and we brought this in contact with either chitin, the colloidal chitin, which is a more solubilized form of the chitin, and then the glucosamine beads. So these are beads where the glucosamine is attached to. So we bring this together, then we spin it down, and then we separate the bound fraction, the B, from the soluble fraction. So if the chitinase is bound, you see here a dense band. If it's still in a solution, you see with the S a dense band. So for chitinase A, you can say that it's not really efficiently binding to the chitin or the glucosamine. It's more in the soluble fraction. The second protein is highly efficiently binding to both chitin and glucosamine because you can't see it anymore in solution. But we're not only interested in chitin, we thought that this is important in mucus breakdown. So what you see here is a dot blot. So that's actually a membrane where you spot the mucus on. Then you bring the chitinases on top of it and you stain the chitinase. So what you see here is two different types of porcine mucus, and you don't see anything, which means that the chitinases didn't bind to the porcine mucus. Then you have 6 different biological replicates of chicken mucus, and you can say that the first chitinase bound to 5 of the 6 chicken mucus replicates. The other one didn't bind. We also made crystal structures of these chitinases, and what you can see here is this is the active site of the first chitinase, and you can see it's open. It's a white active site cleft. So here the really branched complex mucus substrates can fit in, and the chitinase A can degrade the mucus. And this is indeed the enzyme that did bind to the mucus. And we confirmed this mucus degrading activity in the lab, and indeed this enzyme degrades chicken mucus. Then you have the second one, that one binds really efficiently to the chitin, but it didn't bind to the mucus. And here you see it's a closed tunnel, the active site. So a branched substrate like mucus won't fit in, but the highly linear glucosamine repeats from the chitin, they can fit in. So this chitinase is probably not important in mucus degradation. We didn't see any mucus degrading activity in the lab either, but you have those glycans on top of the epithelial cells. These are more linear, and probably this enzyme is important in breaking down these glycans on top of the epithelial cells, but this has to be confirmed. And then what is left is the protein backbone. And for this, Perfringens produces zinc metalloproteinase B, which is commonly found in really a lot of Perfringens strains. And then you have a zinc metalloproteinase that is specific for the NetB positive strains. And as before, we again, the group of Ben Wade did necrotic enteritis trial. So what you see here is a wild type strain where the animals got diseased. Then when they made a knockout in the universal zinc metalloproteinase, you see there's a reduction in the number of animals with lesions, but still it's able to cause disease. And when you knock out the NetB specific, so the pathogen specific enzyme, you see there's an even lower amount of animals that cause disease. Yeah, there's only one that got disease and the others didn't. So you can say that if you target the initial stages of disease, so the breakdown of the mucus, you can also block pathogenesis, and it's not only the toxin that is important. So yeah, you have different steps for the breakdown of the mucus. The first thing is really important is remove the sialic acid, because if the sialic acid is not removed, the other enzymes can't access the other structures. This will also induce a higher toxin expression. Then you have the breakdown of the glycans with various enzymes. One of them are the chitinases. And then the protein backbone is degraded, and the toxins and everything can go through to the next layer. And then it stopped working. So then there was a last slide just summarizing that we have different forms of disease. Maybe NetB is not the only thing to focus on, but there are more things that are also important. And if you want to prevent necrotic enteritis, you should take into account that there's a really complex delivery system to get the toxin to its target cells, and you have to breach the mucus layer first. And that it might be interesting to look at the initial enzymes and initial stages of disease to prevent necrotic enteritis. That's it.
Okay, thank you. Thank you. Thank you so much, Evy. That's a very remarkable presentation, because for me it shows that whatever we thought we knew 10 years or 15 years ago, we always have to question what we were talking about then. So it's a very complex matter, but explained in very clear wording. We have time for one or maybe two questions. I don't know if somebody from the audience has a burning question. Evy will be here also during lunch and also afterwards. So if you have some questions, you can always reach out to her as well. But maybe some burning questions from the audience on this moment? No? Yes? So there's a box, and you will be receiving a box. You just talk within the box and clear and loud, please.
So, hello, Evy. It was very interesting to see how strong the genetic effect was on necrotic enteritis between the fast-growing, the slow-growing, and the organic animals. Last week I've seen an interesting trial how they made a slow-growing bird out of the Ross 308 by feeding a different diet. Do you think it's more genetic or more the type of the feeding which causes the difficulties?
I think it's both, because we know that the animals, there's a really fast cycle of selection of these animals, and you get a lot of feedback from the farmers to the breeding companies. And I have the impression that the standard Ross broilers are selected to be disease resistant for necrotic enteritis. So that's why we don't see it anymore. So there's a combination of the coccidiostats that are used, but also just because of the selection of the animals why they don't get diseased. So I think the other slow-growing and the organic broilers, that they are just not yet selected to be more disease resistant because they didn't know it was an issue yet.
All right. Maybe I just have one additional question, or maybe, yes, there in the back, there's another one. Please, you can hand over the box. Okay.
Thank you for your presentation, Evy. Short question. Do you think that feed additives that stimulate mucus production could prevent the infection by the necrotic enteritis? If the mucus is keeping being produced, do you think that the toxins can't break it down, and maybe then infection won't happen as well?
I think you give more nutrients to the Perfringens and if you have more mucus production. But I think the mucus composition can be really important. So if you have feed additives that steer the mucus composition to something that cannot be used by the Perfringens, it will be less of a problem, because that's something that's not in the presentation, but not every mucus predisposes, and the chitinases don't react with every mucus of the chickens. It's mainly when the animals had a coccidiosis infection, the mucus from these animals, that's where the chitinases react really heavily on. So it looks like the predisposing coccidiosis infection is changing the mucus composition of the chicken, and the Perfringens, the chitinases, are reacting on the change in the mucus composition. So.
All right, clear. I would suggest that we go and move on to the next presentation. There was one more question left, but I would suggest that you reach out to Evy during the break, okay? Otherwise, we'll run in troubles with the kitchen, and you don't want to run in trouble with the kitchen. And you want to have the box. You can hold the box until the next speaker. Okay, quickly then. Quick question then for here on the left side.
I can throw it.
Yeah, throw it. Yeah, okay. All right. Okay, go ahead.
Yes. Highly appreciation for your informative presentation. This from the preventive point of view. How much the using of the organic acids or even probiotic producing the organic acids in the control of necrotic enteritis from preventive point of view and also commercial point of view? Thanks. It's a very short question.
Yeah, commercial point of view, that's not my topic, so I can't say anything about it. But from preventive point of view, I think if you look at the organic broilers, where you can't, well, in Belgium at least, you can't use a lot of additives and you see a high amount of problems. I have the impression if you would be able to give organic acids in the feed to these animals and you can enhance the epithelial barrier and the intestinal gut health, that you would have less problems.
Okay, that's a short answer to a short question. Thank you so much, Evy, and applause again. Okay, then we'll move to the next speaker. This time, Tim Goossens. Tim is, I know him for many years as well. Tim has been studying biotechnology at Ghent University. but he also subsequently worked as an academic researcher at the University of Antwerp, and then he moved to Leuven. So he has been working in the different major universities in Belgium, and then he took up a doctoral project on neurodevelopment in the Laboratory of Developmental Genetics. He then obtained his PhD in 2010. And then he joined Nutriat to work as an R&D engineer, after which he became the business development manager of Digestive Performance, a product range dedicated to gut health and animal performance in production animals. And since Nutriat was, together with FRAmelco, bought by Adisseo, he's now the global technical and scientific director for gut health solutions. And in this regard, he will contribute his knowledge and his insights on a very exciting topic because it starts with size matters, the distinctive effect of short and medium chain fatty acid based products on intestinal health and gut microbiota. Tim, the floor is yours.
Thank you, Maarten. So indeed, I'm very glad to stand here before you and talk about size indeed, specifically size about short and medium chain fatty acids. And what I'll do in the presentation is actually, after reminding everyone what a short and medium chain fatty acid is, I will go to the main three type of things that these molecules can do. So 3 functions. So we have, let's say, an acidifying effect. They can also function as a signaling molecule. And lastly, I will discuss some antimicrobial fatty acids. And for each of these 3 groups, I will raise 3 questions. Namely, like, okay, what from a biological or chemical point of view, what is the starting point, let's say? What are the characteristics that we can make use of? Secondly is that, okay, why does that matter, specifically for livestock production? And then the third step is how, with all this knowledge, how can you further optimize these molecules? And that's then typically a job for feed additive producers. So there they are, listed. As you might remember from chemistry class in school, they're called fatty acids. They're acids because all of these molecules have an acidic function. And then actually they differ only in the length of their side chain. So for example, butyric acid is a well-known one, has 4 carbons in the side, and lauric acid 12, for example. And typically everything up until butyric acid is called a short chain fatty acid. Afterwards, until, let's say, up until C12 is medium chain fatty acids, and from then the long chain fatty acids begin. So there are, I've listed them here horizontally, and actually there are 3, let's say, main groups. So the first 3 ones, you could say, are typically used for the typical acidifier types of product. It can be in the animal, can be in the feed as well, really, let's say, to lower the pH. Yeah? That's their main function. But then we come to a group that is special, because in monogastrics, these 3 are endogenously produced, and the whole digestive tract, actually the whole system of the animal, is wired to react to these molecules. So that's a very different context. And we're talking about gut health signaling. And then when the chain length becomes higher, longer, we have distinctive antimicrobial properties. So it's not because the chain length is, okay, gradually longer and longer and longer that you will have, let's say, also gradually longer and longer, higher and higher melting point, for example. And also the biological function is like, say, a gradient. It's more distinct, actually, as you see. And let's start with the acidifiers. I say the foregut, they can also be used in the feed. But whether their prime mode of action is, let's say, or site of action, the main part is that it's there to do what an acid does best, namely giving protons away. So an acetate, for example, that can split in acetic acid in an acetate and a proton. Actually, you can have full dissociation, but in these type of acidifiers, it's typically important that there is an equilibrium. Most of them are weak acids, so there is kind of an equilibrium between the undissociated acid and the dissociated one. Well, why does it matter? Well, first of all, in feed and drinking water, these acids are very well known to inhibit the growth of bacteria and molds. That's also important for animals in the digestive tract itself, but also, for example, because these acids are important to improve digestion. And typically, as you know, some animals, young animals typically, have a gastric pH which is still relatively high, wiener piglets, for example, so this can be of an issue. And so this is then the context that we talk about. So this is then the acid that you see. You can, with dissociating into protons, you liberate the protons, you lower the pH, and you create an environment in the upper part of the gastrointestinal tract, so the stomach typically, that is less favorable for bacteria, including pathogens. So most bacteria are quite comfortable, let's say, between a pH between 5 and 8. That's why you'd like to push it more, the pH, to the left-hand side, because then they struggle to grow. Unless you're lactic acid bacteria, then you can take more. But it's also important for the digestive process itself. If we think about typical components that we find in the feed, like blood plasma or vitamins or zinc oxide, these are things that go against the typical acidic effect in the stomach. So they have a buffering effect, and we would like to reduce that. Also, there's pepsin there's pepsinogen in the stomach that needs to be cleaved to become active pepsin, and this happens at a low pH. So another reason to have a low pH. If people talk with you about acidifiers, they will often talk about the pKa value. Why? Because it matters quite a lot where this equilibrium is. Because on one hand, it is assumed that this undissociated, or an acid needs to be in an undissociated form to cross the membranes, but once it's inside the bacteria, actually it can dissociate, and then actually you have an acidification of the internal part of the bacteria, but also anions that can have a disruptive effect in the bacteria. So knowing that, okay, what can you do if you know that it needs to end up in the feed? Well, there are things that you have to take into account. So you have to know, okay, are we going for a drinking water product? Is it a premixture product? Do you want to avoid corrosivity, for example? In that case, you might actually better use not only the acids, but also their buffered salts in a mixture. Or the pKa values, where do you want it to be active in the gut? Other things as well. Well, yes, this is an important one. You can put acids all that you want in the feed and lower the pH, but in the end, the animal needs to eat it still. So you have to take into account feed intake as well. And typically, depending on what you exactly want, you will typically end up somewhere, let's say, on a matrix, coming to a specific mixture that would be suitable for you. It's often mixtures, because what you typically like to see is that, let's say, the activity of the mixture is more than the sum of each component. Now we come to acetic, propionic, and butyric. And I said that is typically in the hindgut of monogastrics, obviously, and ruminants, in the rumen of ruminants, swine, poultry, humans as well, by the way. And why is it endogenously being formed? Because in the hindgut we find a lot of bacteria that use undigestible, or let's say for the animal, undigestible substances to break that down, like polysaccharides, into oligomonosaccharides. These will be converted again by other bacteria into these short-chain fatty acids or lactate and other substances as well. There is some quite cross-feeding as well. Some bacteria, let's say, break down and produce lactate, and this lactate is then used as a substrate by other bacteria to produce butyrate, for example. To give you an idea, this is numbers I got from a slide from a publication from Lonneke Onrust, where different concentrations of these short-chain fatty acids in the ceca of broilers were examined, and you see that acetate by far is the biggest chunk of short-chain fatty acids, and you also then have, to a lesser extent, propionate and butyrate. Now, typically, when we look among these three, it's butyric acid that got the most attention in terms of a molecule that can support gut health. Now, why is that? If I just explain you that it's actually acetate who is by far the most abundant one. Well, that's likely to do with the fact that if we look at what happens when they are absorbed, these short-chain fatty acids, well, typically they can be already used and metabolized in the gut, but as everything else, everything that's not used goes to the liver and afterwards to other organs. And that's distinctive, this route between these different short-chain fatty acids. For acetic acid, for example, does not typically reside too much in the gut. It goes to the liver and then for sure other organs like the muscle, this is one example. Propionic is different. Propionic is used in the gut, but then certainly in the liver as well. While butyric is more on the left-hand side. That is, the majority is used in the gut, a little bit is going to the liver, and if there is anything left over after that, then it goes in the more systemic system. So this is probably the reason why if you compare these different short-chain fatty acids and you see what they can do in the gut, so in the gut, not the other organs, but in the gut, typically butyrate comes out, let's say, as the best student in class. And I've selected here 3 examples. Butyrate is, for example, known to induce the secretion of peptides that are antimicrobial, host defense peptides. And there are many, if you measure it, there are many short-chain fatty acids that can increase this or induce the secretion of these peptides, and butyrate is by far the most active. You might say, actually, these graphs are more or less the same height, but it's at a much lower concentration. So at a much lower concentration, butyrate is able to, by far, induce several antimicrobial peptides. The same is true for another important enzyme, intestinal alkaline phosphatase. Same here. If you see C2, C3, C4, C5, you see in this Western blot the thickest band for the butyrate. And last example is that Salmonella can invade cells. So when you put Salmonella together with enterocyte type of cells, then the Salmonella can invade to a certain extent. Actually, if you not only add Salmonella, but also formic or acetic acid, actually you increase the invasion, so you have more. But the opposite effect happens with propionate, and certainly with butyrate is actually the one that is able to reduce this invasion the most. So these functions that I just showed you, it's quite broad, right? I've talked about alkaline phosphatase, about also bacteria, Salmonella, reducing gene expression, and so on. That already shows you that butyrate can do many, many things. It has a wide range of effect. And that is because if we look at the cells throughout the gastrointestinal tract, poultry or swine or human, whatever, you see that these cells have, throughout evolution, built machinery that is able to react to butyrate. So to be able that these cells are reactive to butyrate. This is a kind of a theoretical scheme that I make where all these things come together, but different cells, for example, have different ways in which they can react to butyrate. Well, in the end, you see that it will end up in the Krebs cycle and be used as energy source, but there are many receptors to which short-chain fatty acids and butyrate can bind. If they bind, they can trigger all kinds of a cascade of events. But also inside the cell, they can be incorporated through many active transport molecules. And once they're inside, there are still other things that it can do. It can bind to specific nuclear factors, so cytosolic receptors, or it can really affect the structure of the DNA itself and thereby modulating gene activity. So that's a huge range, actually. Okay, why does that matter? Because when we took at live production animal, livestock animals, then you see that many of these things that butyrate do, the signaling that it does, is that it is able to modulate gut mucosa, immunity, and microbiota. A little bit the holy trinity of the healthy gut, let's say. And to give some examples, I've just showed you the example of the antimicrobial peptides, but it can also reduce inflammation, for example, or increase absorption of water in the last parts of the gastrointestinal tract. Now, I told you these are things that many cells along the entire gastrointestinal tract can react, and can react differently to butyrate. So it matters quite a lot if you add butyrate in the feed is where you deliver it. And you can imagine that has a huge effect. And if you look at the commercial products that are available on the market, you see that by and large the most important one are the fat-coated butyrates and the glycerides, so where butyrate is bound to a glycerol backbone. And if you look where this is released, if the fat-coated butyrates there are where you have a high concentration of butyrates, the fat is there to mask the smell a little bit, but that's it. They will release butyrate in the very first part of the gastrointestinal tract. If it's lower concentration, it quite depends. You have very differences in quality, and most of them actually will release in the beginning of the gastrointestinal tract as well, while others have a more precision delivery profile, increasing butyrate concentration throughout the entire gastrointestinal tract. And then the glycerides, again, are different. They will bypass the stomach or the first part of the digestive tract almost completely because they depend here on the lipase to cleave off the butyric acid, and when they do so, it does so quite readily. Okay, so what are these consequences then? And I give you 3 examples of where we studied these differences in release profile and whether that has indeed some biological consequences. And the first one is actually in stressed young animals, stressed because they got rapeseed meal as their prime source of protein. And what was quite remarkable was that in broilers who got all the butyrate in the first part of the gastrointestinal tract, we saw some negative effects later down in the digestive tract. We saw signs of inflammation in the ileum and the colon, but also of dysbiosis in the ceca. So typically loss of diversity, proteobacteria strongly increased, E. coli, and so on and so forth. So typically hallmarks of dysbiosis, while this was not the case when butyrate concentration was, let's say, spread, because it's the same amount of butyrate, but spread all over the entire digestive tract. A second is quite interesting here as well, because there's still, I think, more to do about fat metabolism. This is something that is typically being studied in humans because of the health reasons of fibers, for example. People know there is a link with fibers, more of this bacterial growth, and it's clear, for example, that people who eat a lot of fibers, their insulin sensitivity is higher. So this a big topic, obviously, and it plays the short-chain fatty acids clearly play a role in there as well. For example, so in the hindgut, if you eat fibers, short-chain fatty acids are produced. This propionate, there are some, if you go to the portal vein, there are some sensors for propionate there that will induce some signaling that goes to the brain. And from the brain back to the enterocytes, where intestinal gluconeogenesis will be stimulated. Butyrate does that so directly. So butyrate here in the enterocytes can stimulate the production of glucoses. Now, why is that important? Because that will go back, that signal, to the brain. If there's some glucose sensing, glucose as well will be sensed in the vein going to the liver. Will induce some signaling to the brain, and the brain will react to it, so this famous gut-brain axis here, in lots of ways, for a lot of organs. It will drive the metabolism more towards expenditure, energy expenditure, towards more oxidation of fat, less storage of fat, increasing insulin sensitivity, and so on. And this is a very interesting topic, because if we look at it from the livestock production animal side of things, if you put that next to some research data like this one here that was published in 2017, and where different kinds of glycerides of butyric acid were given, does not matter, it's different combinations or on top of each other or not, does not. If you look at tributyrine and monobutyrine mixtures, all of them have less percentage of abdominal fat. So here as well, you see this effect that indeed there is less storage of fat and more oxidation of fat. And that can be really interesting because you can, well, first of all, make more muscle, more protein, but perhaps it can be interesting as well if we think about diseases, about fatty liver diseases. Last one is about salmonella. And here we took the PhD work of Lonneke Onrust and Pierre Moquet, where they really looked at different types of butyrate, including ones that are not commercially available with a very strong wax coating, very, very resistant, sometimes with starch added to it to loosen it up a little bit again, to really deliver the same amount of butyrate in different places, and then see what happens. These, if they were protected well, these butyrates, they were able to increase butyrate concentration in the hindgut. Probably, by the way, not all directly, but indirectly as well, but that's another set of data. But also, if you look at the relative butyrate concentration, which is also very key to gut health, how much butyrate in the total short-chain fatty acids? And then you see there is some very nice trend that the better the protection is, you say, the more you go to the right, the better the protection is. Eh? So the more you go, you bring it in the hindgut, the higher the relative butyrate concentration is. Now, why is that important? Let's bring in, like say, okay, this is the x-axis. Let's bring in the y-axis. The y-axis is actually the number of salmonella that you find back. This was a salmonella infection experiment. So the broilers were infected, but then you do see actually that you have a linear, almost linear decrease, that the stronger the protection is, the more relative butyrate you have in the hindgut, but also the less salmonella you can find back there in the ceca. Okay? Looking at the medium chain fatty acids, then we typically use the word antibacterial, although I added it does other things as well, but today here I want to focus on the antimicrobial properties. And why is that found? It stems from the 70s, where it was found that mother's milk must contain some antimicrobial fats, and then a whole kinds of research came up to that with lots of PhD students testing all kinds of different fatty acids and fats and derivatives of these fats, and you combine it with a whole list of bacteria, let's say, with your pathogens of choice, and you see what happens. Are these fats antimicrobial? And they found out indeed that they are, and most likely because, of course, when the chain length becomes longer, you have this very interesting mixture of length and hydrophobicity and hydrophilic characteristics that become very well in disrupting the bacterial membranes. So you can sort of insert it and wreak havoc there. So in the extreme events, you can really have lysis, you can form pores, but also, for example, if you disrupt the membrane, then you also disrupt the enzymes that you can find in the membranes and that are very important for energy metabolism of these bacteria. And if you disrupt those, then you have an antibacterial effect. Now, for livestock production, it matters, obviously, because, well, we do have some well-known gram-negatives and gram-positives, E. coli, Brachyspira, Lawsonium, Clostridium, what we just talked about, and they have the potential to have a bacteriostatic effect on these pathogens. I mentioned viruses, but I grayed it out, because in theory, viruses, there are some viruses with a fat envelope around it, and in vitro, you can show that they have this antiviral effect well. In vivo, there are some interesting results as well, but that has more to do probably with an immunological effect that you induce. But that's a topic for perhaps another time. So what comes out of all these tests? Well, first of all, if you really can play with the length of the because the C12, when you look at gram-positives bacteria, it's more or less clear that C12, also C10 sometimes, but C12 for sure comes out as the best in class. Not against gram-negatives. There, C12 is less effective. If you think about certain Enterobacteriaceae, for example, there the C3 to C10 are more effective. The glycerides, that's a second thing you can do. If you add a glycerol backbone, and you make sure that you have to do this synthetically, but that you make alpha monoglycerides, then they are even more antibacterial. So alpha monoglycerides of lauric acid are even more antibacterial than the free lauric acid. And then actually, if you have mixtures of these mono and triglycerides, then you have some additional beneficial effects as well. These are some examples here from growth curve analysis that was performed, E. coli, typically gram-negative. As you see, well, I don't say this is the control. I don't say the FRA-C12, sorry, the lauric acid does not do anything. The glycerides, it has some effect, but you see that the C6 one actually has a stronger effect. For gram-negatives, because for the gram-positives, like Enterococcus faecalis, for example, it's clearly that only the C12 glycerides were effective in this test. This is, by the way, something also we are investigating. We are taking it out of the in vitro phase and seeing some very encouraging results that also ex vivo, if you take some content from the broilers, for example, that you do see some of the same principles coming back, that you build up a gut, let's say, that is more resilient against these gram-positives if you give them these C12 glycerides. This is another example about Streptococcus suis is a positive one. Yet an example here, if you look at the MIC analysis, then you see that the monoglycerides of C12 have a lower MIC value than the free ones, and this is the basis of testing that we are doing with Streptococcus suis in the field, in challenge experiments at the Schothorst in this case, where you do see that in the treated group, based on this C12 glycerides, that you're able to reduce the Streptococcus suis in the tonsils or in the ilia. Now, that was the theory, at least, a very interesting part, but I'm sure you would like to know what this means for practical reasons. If you think about the challenges that you're facing when raising animals. And I listed here just a number of them, both in poultry and in swine. You might think, or you might hope perhaps, that it's just a matter of sorting things. That you say, okay, we've talked about now these different fatty acids from short to long, and then, okay, the short ones are perhaps, yeah, I just did it randomly here, that you say, okay, perhaps the pure acid is against one disease, but if you take the salt of it, okay, then it's another one. Butyrate, if you coat it, then it's the best one, or if you have salmonella issues, then the coated one is the best, but if you look at eggshell quality in laying hens, then you have to go for the glycerides, for example, and the same thing, and perhaps sometimes you have to combine it. So that would be the thing that many people, certainly salespeople as well, would hope for, a very clear, nice story. And it would make, indeed, my life much, much simpler if that would be the case. But it's not. The reality is that we have to go back to the gut health. And we have to realize that many of these diseases or challenges, they have a common theme. They have the common theme that like again, the holy trinity of gut health, that they are disrupted one way or another. So if you talk about the mucosa, you have integrity loss, microbiota, you have dysbiosis, and the immune status you often have an over-inflammatory reaction, which can be disruptive. And what these short and medium chain fatty acids do, and by the way, other additives as well, probiotics, phytogenics, they all in different ways, but they come together, let's say, in improving this gut health, and this gut health, okay, then will be better gut health will help to mitigate the negative consequences of these diseases. So it's not a one-on-one. It does also not mean that whatever you do, well, it does not matter as long as you improve gut health somewhere, it will be good. No, you have to do a little bit more of an effort. You really have to look like, okay, per case, in specific cases, for specific producers, what are the issues? What have they experienced with? What are they using now? And then look to which one of these molecules can bring the most value in bringing a complete solution. And I give you here some examples. These are real-life examples of customers that we have been talking to and that we have been discussing with. And as again, it depends really more on the specific situation rather than on the challenge. If we look at broilers, for example, we have this case where an integrator was interested in using butyrate, wanted to look at different options, and in the end, was looking to a way to say, okay, we use a precision delivery coated butyrate in the starter period, but I also want to use something during the grower period because there are some issues with dysbiosis. Problem is, of course, these animals are getting older, they eat more, and then actually they want to switch to a glyceride. Why? Because in these glyceride there were some antibacterial monobutyrins inside of it as well. But also, of course, you are able to deliver more butyric acid at a relatively lower cost if you do it per percentage, which becomes more important if your animals grow older. Another case could be where we looked at someone who was interested. It was already using a probiotic with good success. It was improving performance. It was improving the challenge that they had with the Clostridium perfringens related dysbiosis, but they want to do something extra, specifically for the Clostridium perfringens. Now, that is a gram-positive. As I told you, the glycerides of lauric are the most active against one, so okay, there was supplementation in the starter and the grower phase of lauric glycerides. A last example is someone really interested with the veterinary practice to look at ways to prevent BCO, so lameness in birds, in older birds. And they say, when I'm using lincomycin in the young birds, I do see, only in the starter phase, I do see that they have less BCO issues later in life. Probably because already there is some bacterial growth of Enterobacteriaceae, there is some leaky gut, they translocate to the joints, and when the animal becomes more heavy, okay, that's what the problems begin. So if we can tackle that thing in the beginning, that would be great. And yes, there are many things you can do. You can do about the leaky gut. You can do about the bacteria. There would be many of these short-chain fatty acids and other products that in theory could help. But this one was most attractive for this specific case, where we say, okay, let's take the glycerides, but a whole range, against the typical gram-negatives and gram-positives, Staphylococcus, Streptococcus, E. coli, that are typically implicated in BCO. Let's take a broad range. In swine, some examples, let's give, of course, if you talk about, okay, zinc oxide ban, that in a few days, I guess, becomes official. Many things that you could do, many things that could be argued to be work for that, but in this specific case, with this customer, we were discussing, okay, let's work with lauric glycerides, in this case in the late gestation and lactation phase, because there are quite some data that show if you give it to the sow that their piglets actually will perform better, so they're off for a better start. And then, in this case, come up with a concept where you use glycerides of butyric laurit and lysolecithins as a more of a complete concept. This one, maybe we give one more example. I'll use this one. This was specifically for demedication. It was in Spain, weaning piglets. They were already using a butyrate, in my opinion, the wrong one, but okay, we have to be mild. But they were already a butyrate, they want to be happy with it, they want to do something on top of it. And specifically because in Spain you can only use nowadays one type of antimicrobial in the weaning. You cannot combine. So it's a little bit of a gambling work. So they say, okay, we would feel much more comfortable with some kind of a broad spectrum antimicrobial support. And that's indeed where this mixture comes up again with C3, C4, C8, and C10 glycerides. Let me go to the conclusion. So yes, coming back to the title, size does matter for these molecules because, as I've shown you, they have very distinct effects. You can almost group them, let's say. They have very distinctive effects and also potential benefits. I say potential because what you get out of that potential actually, that depends on what you do with them. And that can be working with, when we think about acidifiers, with working with mixtures of acids and salts, or when we think about butyric, that can be coating or glycerides, or that can be working with some combinations, antimicrobial combinations, as we talked about. So many things that you can do. the trick is, rather than saying, okay, this product is for that, you have this problem, try that, and you will be fine with that, it's actually what it requires is that you take the time to investigate really about, okay, what is your specific situation, and what would fit, what would be for you, in your specific case, the best solution. What is actually what you would need? And finding for that specific, the most valuable thing that you can use is, of course, a discussion where we would be very happy to have that with you. And with that, I'd like to thank you for your attention. Thanks.
Thank you. Perfect. Great overview of, let's say, a lot of different type of concepts and products. So you did that very well in time. We are a little bit behind with the schedule in general, but we still have some time for one or two questions. So I don't know if somebody from the audience has a question for Tim. Not at this moment. And I have a question for you. When you look at the multiple amount of products, commercial products, and I think Adisseo is a very nice example, because you have been, let's say, bundling forces with different companies with different backgrounds. What I struggle with, let's say, as a consultant or people supporting people that have to make decisions on what should I put in my drinking water or in my feed, is how can I make that choice? Because there's a multiple of approaches. They all start to make sense, right? I think in the past we didn't always really understand how they link to each other, but now we start to understand really well how they interact with each other. But how can you make a choice of which ones to choose in the end and at what levels they have to be put in the feed or drinking water? How do you make that decision?
I think you have to be in the interesting area in the middle. So on one hand, you cannot say, okay, what is the best short or medium chain fatty acids, and use that for now on and only advise that. The other extreme would also not be fair. That you say, okay, we have the entire list. What do you like? You're more of a phytogenic guy. Okay, no problem. We have a phytogenic and do that. It's actually in the middle where you have to be. And we see there that there is some common themes come back. And if you do a lot of research, you do see not all the trials go the same way, but you do see some patterns coming back. That will be able to help you. So based on the trial data that you have. And on the other hand, also the experience of the user. Sometimes it can be very different. Where people know for sure that, let's say, one type of product or a phytogenics are the prime class in that example. People really like it and they like the concept and they see good results with it, or not at all. They say, okay, no, this is just to be, the witch with adding a little bit of pepper and salt. But if you make the choice and if you really talk about, okay, if you bring everything together, it becomes much more simple. From a theoretical point of view, things stay very broad. But if you look, okay, what are you using? What is successful? And what room is there still for you to use? Because we also have to look at the financial aspect, of course. Then we can, let's say, make an educated decision on what probably would be the highest chance of success, I think.
Okay, clear answer. Anyone else on this moment with any burning questions? No. If not, I would like to invite you all to join us for lunch. Tim and Evy are still around for the whole day. And we will be back at 2, so we have a little bit more than half an hour for the lunch, which is just next door. Okay, so I'll see you back at 2 p.m. See you soon. Thank you.
When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something, yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something. Yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice for our animals and our products, but also a healthy choice for ourselves. And when it all comes together and all just works, this is the result of a healthy choice between a clear vision, a bit of elbow grease, and a deep passion for excellence. Together we can leave a world behind a little bit better than when we found it. We're all passionate about something. Yet not all our needs are the same. Sometimes good needs to be better, and sometimes better needs to be outstanding. When we run into obstacles and challenges, it's good to know we're not alone. There are others like us that have the same goals and can alleviate some of our burdens. When we meet others that have the same standard and passion, and that are also inspired to discover the answers alongside us, we can all truly grow. When we strive to become a healthier business and work alongside a partner that shares the same ideals, we not only make a healthy choice.
that lunch and that lunch break, we have to continue for the next session. And as promised in the introduction earlier this morning, the next session will focus more on the nutritional side of things. And we will have as a first speaker, we will have Francesc Molist. He's let's say, a good bridge between the two topics, because he is a veterinarian and a nutritionist. I didn't know they actually exist, but okay, that's a rare specimen that you will see in front of you in a minute. So he joined Schothorst Feed Research as a researcher and consultant 11 years ago, in 2011. And since 2017, he's leading the research group as research and development manager. He is coming from Barcelona, the University of Veterinary Medicine in the Autonomous University of Barcelona. And during his master's and doctorate in animal science, he collaborated with several major research groups in swine nutrition from across the world. Yes. So Francesc will talk about fat energy and digestion in monogastric animals. You have 45 minutes, and then we will have some time for some questions. Francesc, the floor is yours.
And first of all, thank you to FRAmelco first and second Adisseo for the invitation. I'm Francesc Molist. As Martin said, I study vet, but I never work as a vet, but still I know something on physiology and let's say disease. But mainly I've been focused on, as nutritionist. So as you will see in this presentation, I will mainly focus on fats and digestion from a nutritional point of view, let's say, and then, if you have comments or you don't agree on something, let me know. When we go to the, let's say, the fat wall, as you will see for the nutritionist, is not easy. So we have many different products. We have, let's say, not only as ingredients, but also additive, different, let's say, solutions. So I will try to simplify and focus on the ingredient and energy and some effects, but we can extend it as much as you want. So this is the let's say, the agenda of my presentation. I will focus first a bit on the composition of some fats and oil to see some values, some characteristics, then we'll move to the digestion. And then, so the first part will be mainly related to the energy value, and then we'll move more to functional ingredients. So we have seen already this morning that fats are probably not only giving energy, can have also some functional properties. So I will try to also give some insights from a nutritional point of view. So if we start, let's say, on maybe this have changed in the last month, but this was a typical sheet to understand, okay, what we have available as animal nutritionists when we look to the oils, let's say. Most of the oil still was coming from the palm industry, so palm oil was one of the abundant one. Then we have the soya oil, then the rape, sunflower, and then you see some minority oils, let's say. This could be that has changed in time, let's say, because there's, as you know, there's a huge discussion on the sustainable production of palm oil, let's say, and this probably, in the future will bring some challenges for the feed industry if we don't have this available, let's say. We need an alternative, and this, I think, we need to take this into account, that maybe due to sustainability issues this can go down, and so we will see how it goes. If we look to some compositions, these are table values that you can find in the CBB or other places. Here you have the different oils, let's say vegetable oils, and here you have the fatty acids. So you see, I put in red what are the main differences. If you start with the soya oil, you see that is mainly rich in C18-2. When you go to linseed oil, you see that is mainly rich in 18-3. Rapeseed oil will be mainly rich in 18-1. Palm kernel, as you know, similar like coconut oil, is mainly rich in the medium chain fatty acids that we were discussing this morning. And then you see the palm oil that is more the C16-0-18-1, and one ingredient that is more common in Asia and less in Europe, let's say, will be the rice bran oil that you see also is rich in 18-1 and 18-2. These are the vegetable oils. If we go to the, let's say, more the animal fats, here you have also the same, let's say, fatty acid profile for the different, let's say, ingredients. Then you see the fish oil is more on the 18, 20, 22. So here you will see the fish oil that mainly plays a role on the functional property, let's say. And then you see the different fats coming from animals that also vary a bit. So the tallow will be mainly C18-0, C18-2, the let's say, will be higher in lard and even higher in poultry fat. And then you have all the mixtures that you have available in the market in many countries. They are blending different fats. And then it's good to understand what you are using and try to at least understand what is the fatty acid profile in order to get the right energy value. As nutritionists, we say, okay, we want to understand what we are using. We want to characterize the fat that you are using, so you can do quite a bit of analysis. No? First, you could start with the, let's say, the physical appearance, taste, smell, to understand what is rancid or not. Then you can go to moisture, impurities, the iodine value. So you see quite a bit of analysis available. And probably, as you will see in the future, that we have more techniques available and we want to really formulate real time. Probably not only the crude fat level and the iodine value will be important, but also the free fatty acid and even the fatty acid profile. So some of the, let's say, companies are already not only working with the crude fat level, they are already entering as input the fatty acid profile. And this, I think, will facilitate the work of a nutritionist. So I think with NIRS and other techniques, we will really be able to get more understanding on the fat that we are using, what is the quality, how we need to use, and this will help, for sure, the nutritionist. So if we look to some of the analysis, just to show you some examples. So here you see the moisture, impurity, peroxide, free fatty acids, total fatty acids, and the rest fraction. And you see here the rest is when you take everything out, and then the UR ratio, that is the ratio between unsaturated and saturated fatty acids. And then you see some values, but what is interesting is that the rest in some of the fats is quite big. So, and that means that, when you call it rest, that means that we don't understand what is there. It can be waxes, it could be other sources, let's say, but you see that for some, still the rest fraction, is quite big, so we need to better understand what is in there, and especially as you will see, the free fatty acids will also be really important. And here you see, for example, this free fatty acid mixture that has around 70% of the let's say, of the acids are free. And you will see that this has consequences for the digestibility. So understanding how much of the acids, let's say, are in the free form and how much is still with glycerol, and this is really important to predict the digestibility. But still, as you know, we have work to do in understanding what is in this rest fraction and how we can characterize it better. Then the next step is to give an energy value. Nowadays, with the current situation, everyone comes with matrix values, and then the nutritionist needs to understand, okay, I'm doing the right, I'm using the right levels or not. But here you see again some, let's say, some table values for total fatty acids, an example of free fatty acids, let's say 100, or this mixture that I was mentioning, the UR ratio, the gross energy, then the digestibility, and this you can debate, You can say, okay, is true that, for example, the palm kernel fatty acids, let's say, is only 81% digestible or is higher. These, I think, are discussions that we have nowadays on ranking the acids. And then you get a metabolizable energy or a net energy, depending if you work for poultry and swine, let's say. And here you see this still you can correct, let's say, on dietary level, if what is the UR ratio at dietary level, and you can still uplift the energy value of some of these fats if you want, let's say. But I must say that the discussion on the ranking is still, although we are using fats for a long time, still is a current discussion how to characterize the fat, and probably we need to work, to do a bit more work. The main challenge on fats is that when we do digestibility trials, you want to include the ingredient at a high level in the diet in order to measure the digestibility, but you see that in fats you have an effect of the inclusion of the fat. And when we do digestibility trials with protein sources, for example, we mix a basal diet with 20% soya bean meal or another protein source, and then by the difference method you calculate the digestibility. In fats you see that the level has also an effect on the fat digestibility, so that means we do less digestibility trials in fat that we should do, let's say. Then if we go, so this was the characterization, then we go to the digestion. And then as let's say we discussed this morning a bit, you see that the fats will be different digested and absorbed depending on the size, no? But mainly in the small intestine will be the absorption. So first the digestion, then will come the absorption. And this degree of absorption will depend on the animal first, no? You will see that it's a different piglet than a growing pig, or a broiler than a piglet, for example. You will see that there's some interaction at dietary level. So if we have higher level of calcium in the diet, we are penalizing, for example, the digestibility of the fat. So that's good to take into account. UR ratio, very important. As this morning was present, the length of the fatty acids is very important, medium versus short. The presence of monoglycerides, emulsifiers, the position of the fatty acids is also very important, and also probably the particle size and a bit more on the technology. So it's not only the ingredient that is important, it's also many factors will play a role how we, let's say, predict the digestion and the absorption in the animal. So as you know, the digestion starts in the stomach, no? There's this lipase, and when you feed a triglyceride, the first step is to start cutting down to these monoglycerides and fatty acids. So this is the first step. We need the lipase. And the second step, when we have these, let's say, free fatty acids and the flat globules, then the bile salts play an important role. And I think, let's say, we understand quite a bit the digestions of fat, and we understand the effect of lipase, and we are interested on the effect of lipase. But you will see that when we have, for example, a challenge and the bacteria are growing, then the bile salts disappear because are taken over by the bacteria, and then you will see one of the problems why fat digestibility is reduced is not because of the fat is not available or whatsoever, it's more the bile salts that are not there. And that's probably when we have a challenge, we really need to be careful that the bacteria don't grow, because if the bacteria overgrow, then you will see that the bile salts disappear, and then the whole process of digestion is impaired. Then you have the medium chain fatty acids that you don't need to form let's say this figure, let's say, and it will go more on diffusion. So when you have a challenge situation, medium chain fatty acids will always be easy to digest than the long chain fatty acids that we need to form this micelle. So this is a bit to explain now. We work more and more on diets free of antibiotics, on diets without zinc oxide. So that means that in the future, the microbiota or microflora will play a more important role. So we were used to, in the past, to control the microbial growth, and then the rest of the digestion was good. In the future, we need to deal with the bacteria, no? They are there, and that means that the interactions between bacteria and other components will be very important. So again, for fat digestibility, if the bacteria grow, then the bile salts disappear and the whole process is impaired. And then if you look in the feces, you see that the first nutrient that will increase, let's say, in the feces is fat, because the digestibility is really under challenge, let's say. So this is an example. Some time ago already they did this trial, but here you see clearly this is the relation between bacterial growth. So this is in vitro, but you see the relation between bacterial growth and the conjugation of these bile acids, and you see the effect is linear. So more bacteria is growing, more bile acids are used, and fat digestibility is dropping. So if we cannot stop this growth of bacteria, let's say, the conclusion will be fat digestibility will go down. And in this case, this is a trial that they did more fundamental for broiler, but will be the same for swine, let's say. These are animals with what they call a conventional raised animals with microbiota inside the animals and a normal bacterial population. And this is what they call germ-free, so no bacteria in the gut of these animals. And then they were feeding a normal maize soya diet, or what they call a maize with some a component that could increase the viscosity. And as you know, viscosity in broilers is a problem. If you increase viscosity, the whole transit stops, and then bacteria can grow, and then you get dysbiosis and you get problems. So you clearly see, in this case, is the fat digestibility. When you don't have bacteria in the small intestine, let's say, of these birds, you see high fat digestibility. When you have normal birds, let's say, and you increase the viscosity, then you see the fat digestibility decrease really a lot. So let's say when we have challenge situations, we really need to preserve the fat digestibility, because we can include as much fat as you want in the diet, but if the bacteria are growing and they are using these bile salts, the absorption will be compromised and the bacteria cannot use this as energy source. Then we say, besides, let's say, the bacteria, as nutritionists, we say, okay, we need also a tool to try to understand how we need to formulate diets and what is the relation between, for example, in this case would be the unsaturated-saturated ratio, let's say, and the digestive absorption of the fats. And here you see two scenarios. You see a piglet and you see a more adult animal, a growing pig, and this work was done by the PhD, let's say here, and you see that in young animals we say the UR ratio should be more or less around 3, and then you see that the fat digestibility is already above 95%. In adult pigs, growing pigs, you could even go lower, 2.5 if you want, and you see digestibility is good. And then you see that if the digestibility is below 2, let's say, you see that the fat digestibility is really compromised. So as nutritionists, we can already, when we formulate diet, take into account this UR ratio at, let's say, feed level. And with this UR ratio, we will already understand at what level we have our fat digestibility. And if we are in this scenario, then probably you need to use emulsifiers to try to increase the fat digestibility. If you are here, you see already that if the animals are healthy and everything goes good, you are already above 95%. Then, as I mentioned, let's say the animal is also really important. So in Schothorst, we try to understand, for example, what is the difference between a piglet and a growing pig. And in this case, the differences are based on fat digestibility. And you see, for example, the piglet is the red bar, and the growing pig is more the blue. And you see that the piglet digestibility of fat is always low. But you see that the ranking is not the same between piglets and growing pigs. So that means that nowadays that we say we want to kick out zinc oxide from our diets, we want to remove antibiotics, then probably we need to better understand, in this case in piglets, how much is digested the fat in piglets in order to give the right digestibility values to those piglets. This is a trial that we did to understand really the effect of this unsaturated saturated ratio. So here you have trials, in this case, growing pigs, and you have two different compositions. So you have, let's say, different treatments, one only with soy oils, and then some fatty acids, and also soy oils with some more free fatty acids. And then you see the UR ratio at the feed level. So you see with soy oil, you are above 2.5 all the time. So in principle, in this scenario, you say no big problems, let's say. When you are using palm oil, for example, then you see that the UR ratio is going down. So if you remember the graph that I made, in this scenario, the fat digestibility will be lower than in this scenario. And so then if you compare then the effect of soya versus palm, you see a reduction, but this kind of soy oil is more digestible than palm, this we know. But when, for example, we have more free fatty acids, then you know that the differences are really big. So let's say the worst situation is that when we have a really low UR ratio and more than 60% free fatty acids, let's say, then you see that the fat digestibility is compromised. And so in this scenario, probably we need to find an alternative solution, and probably emulsifiers can be a solution to try to go up again on the digestibility, because if not, you see the drop on digestibility is really big. Seems doesn't want to go to the next one. No. If you do the same for broilers, so the same setup for broilers, so here again, soya oil as only fat source, then soy oil with some free fatty acids, let's say, and only free fatty acids, and the same for palm oil. Here you see again the UR ratio, so for broilers will be the same situation. Let's say above 3 or 2.5, you are safe, below is a problem. And then you see here also that if you compare here, of course, soya higher than palm. When you have some free fatty acid, it goes down. But again, the worst scenario is a low UR ratio with only free fatty acids. And then you see that the digestibility drops from, let's say, 80 to 52. So you really need to control the amount of free fatty acids that you have in your diet. You need to control the UR ratio if you don't want to be in this situation. So these are fat sources. As you know, as nutritionists, we also have and will have more byproducts that still have some fat inside, let's say. So you see a lot of oil cakes, let's say, available for the feed industry, and then you have a combination that is still there's some protein, some fiber, and some fat. And what is, let's say, important is that in these byproducts, still the fat is in most of the cases inside the legume, let's say. So it's not really easy available. So if in the future, if we believe in circularity and all these concepts, and we get more byproducts available, like DDGS, for example, DDGS in Europe still can have 15% fat. Let's say if you go to US, only they have 3%, but in Europe still the fat content is quite good, let's say. So in this scenario, we will probably need to better understand how in those ingredients we need to process them in order to get the right energy, let's say. And this is where comes, for example, the effect of processing. So this is in swine, when we are giving a meal diet, so not pelleted, versus a diet that we are pelleting, let's say, then you see clearly, and this is mainly in rapeseed meal, but it could be, you will see it in a dietary level, then you see that the biggest improvement is on the fiber, of course, because by pelleting we make it smaller and more available for the bacteria, but you also look in fat digestibility, the increase is substantial, let's say. And this is again the combination of making let's say the more small, and also making the fat more available for the lipase, for the digestion. So you see, really depending on the byproducts that you are using, if you stick to meal, still, you are not usually using, let's say, the full potential of the ingredient, and by pelleting you can increase quite a bit. This is for rapeseed. If you say, okay, at a dietary level, maybe the batteries are going to... No. If you look to the same at diet level, then you see the same. Meal more the dark ones, and pellet diet is more the light ones. And then you see in organic matter, protein, no big difference. But in fat, you see this should be here, there's a significant positive effect of pelleting, and the same for the fiber fermentation. So pelleting improves the digestibility of fat and fiber, and of course, depending on the animals, in piglets probably protein will also play a role, but especially fat and fiber are the two nutrients that most benefit from the pelleting. This was, let's say, from a nutritional point of view. Now we move to what we saw this morning a bit as functional ingredients. No, fats has an energy, but also we can use it as functional ingredients. We start, for example, with the omega-6, omega-3, no? And this is a ratio that maybe, let's say, 5 years or 10 years ago, nobody was, or not many people were paying attention. Now again, that zinc oxide needs to go out, and we have much more relation between bacteria and nutrients. People are, let's say, trying to understand what is the optimum ratio. For humans, there's quite a bit of studies done on the omega-3, omega-6, omega-3 ratio. For animals, less. But in principle, we say, okay, animals, they cannot produce. They can elongate, but they cannot produce themselves, let's say. And then what is really important, as you know, is that the omega-6 are mainly pro-inflammatory and omega-3 are more anti-inflammatory. And it's not that we only want one or the other. The ratio, as you will see, is very important. And both are these polyunsaturated fatty acids. This is how it looks like, no? As you know, the different acids, so they are more or less similar, only you see here a big difference, let's say. But as you will see, the ratio of both is more important rather than saying only one or the other. This is the classical graph that you see. Animals, they cannot produce, but they can further elongate, and they arrive to this DHA fatty acids that we say, okay, this will be the polyunsaturated fatty acids that will have some anti-inflammatory. So the green ones will be more on the anti-inflammatory. These will be more pro-inflammatory. And in humans, in animals, I don't know how important it is. They even say that we are more intelligent if we eat more fish and we go to this direction, let's say. So it's not only the inflammation, there's also more, let's say, metabolic effects rather than the pro and the anti-inflammatory. Then where those acids, fatty acids come from, typical ones were coming was the fish oil coming from the different, let's say, fish. This is, let's say, a table that maybe is already outdated, and the main reason is that nowadays we are using also for fish quite a bit of soy oil, and it makes that the fatty acid profile of the fish is more similar now to vegetable oil than what was in the past. Let's say in the past when you had wild salmon, it was really rich, let's say, in EPA and DHA. Now these values are more under discussion, and it's always good if you want to use this fish oil to analyze once in a while what is the fatty acid profile, and I'm really paying for these acids, let's say, or what is the content. But this is changing. On the other hand, besides the fish oil, we have now other oils coming in the market. For example, algae oil is also really rich in DHA and EPA. So in the future, we also do not need to be dependent on the fish oil, and we'll have probably some other oil available that will bring more this the C18 and the C22. When you look to the effects, why we need to control those acids, so this is a trial that was done in piglets, and here what they did, what is important, they had two different conditions or farms that they say, okay, really good health status and poor health status, and then they were sacrificing the animals, some animals at day 7 post-weaning, some animals at day 14 post-weaning, and then they were also looking to the diarrhea, and diarrhea as a measurement of gut health, let's say. And here you have the ratios between omega-6, omega-3. So starting from really high, 21, to more low, 14:1, let's say. And as you know, this will be, if you go back to this graph, this will be more pro-inflammation, or the higher, let's say, these omega-6 will be more inflammation, and if we go down, we go to more anti-inflammation. And then if you go back to, let's say, to the effects, here you have the effect of the, let's say, environmental conditions, clean versus no clean. So here at day 7, no effects, and at day 14, you see there were all effects. But when you see the effect of the ratio or the interaction, then they didn't see any differences on the gut morphology, so they could not see, okay, more, let's say, this lower ratio, longer villus height or more creep depth. This they could not see, but they found a significant effect on the, let's say, on the diarrhea score. So then that means that probably there's something in. So if you say, okay, where we need to pay attention on the ratio, probably I will start working on piglets, especially when you want to take out zinc oxide, antibiotics, there you can play a role. And probably also in lactating sows, because the metabolic rate is also quite higher, so they probably can play a role. In growing pigs, I could probably pay less of attention. Then we go to the other functional ingredients that will be the short and the medium chain fatty acids. This morning already they explained quite a bit, so I will not go too much in detail, but as this morning was mentioned, from the short, the most interesting one probably is the butyric acid as a source of energy for the enterocytes, let's say. If we go to the medium chain fatty acids, we talk mainly between the 6 and 12. We have it as additive, no? And also this morning they present quite a bit the different solutions, and we have it also as ingredient, and then it's a matter of cost, availability, and what we want to do to say, okay, I go for one or the other. Compared to the long chain fatty acids, the digestibility is much higher, no? Because they don't need to form the micelles, the absorption is go really directly inside, so it's less complex. But of course they are shorter, no? And if you look to the gross energy level, is lower in the short versus the long. And then, of course, because they have these gut health promoting effects, then you can also give a bonus or an energy value to these gut promoting effects. And then, as we saw this morning, probably there's some opportunities to combine the short with the medium, depending on the problem that you have to tie through to have a more broad, let's say, approach. Especially in young animals, probably is where there's more opportunities. Those animals are more immature. We get more interactions with the bacteria. They have more challenges. So if we can control this challenge and we can avoid the diarrhea, or we can avoid the wet litter, probably, we will benefit from controlling this, let's say. This is a trial that we did also in Schothorst quite some time ago. So in this case, we were using piglets post-weaning, and we did a trial for 4 weeks post-weaning. And the focus of this trial was on the fat source. So we are using mainly animal fat, coconut oil, that will be this medium chain fatty acid approach, soya oil, let's say. And here you see for the total period, feed intake, average daily gain, the feed conversion ratio, and the fecal score. The fecal score, you see a value of around 7. So this means no diarrhea, because the higher the fecal score means more dry feces. But to see diarrhea, we probably need to be around 5.5, let's say. So there was no diarrhea in this trial, so the health status was good. But still, you see, with the coconut oil, we saw the most promising effects in terms of feed intake, average daily gain, but also you see the FCR was also better. So sometimes you get the discussion in the field, hey, be careful with the medium chain fatty acids, that they have some problems with the taste, because the taste can be quite unique for those, and the piglets will not eat. in this case at 4%, and this, with the current feedstuff prices, this will be an expensive diet. 4% coconut oil in a piglet diet. Still, we saw, let's say, positive effects, mainly coming from feed intake, but also they were growing more, so even the efficiency was better compared to, let's say, the golden standard that will be the soy oil. This again a bit on the, let's say, on the link between the medium chain fatty acids and the different bacteria. No, I think already this morning we understood that we don't have, of course we have solutions for everything, but we need to understand which problem we have in order to find the right solution. And I think this, in the past with the zinc oxide, with the antimicrobials, a lot of companies were able to produce one diet for the customers, and they were saying, okay, this is my piglet feed, and I want to use it in all my customers, let's say. You want to change? Yes. And what you see nowadays with all the challenge that we have around, I think the future is impossible to have one feed range for your customers. And I know that is difficult for the plant manager and for the logistics and everything, but the future goes to understand what customers do you have, what are the challenge, and depending on the challenge, you need different solutions. And as this morning was discussed, if you have gram-negative bacteria, you'll need other solution than when you have gram-positive bacteria. And this is, I think, important to understand, to make the right decisions, to say, okay, I have a farm with this problem, for this problem, it makes sense, this solution. I have another farm with that problem, let's look for another solution. Universal solutions, I think it's difficult to get them. Oh, we didn't improve much. No. So the new future problem, as you know, the people in the Netherlands, but in Europe, will be a Streptococcus suis. So diarrhea, I think we have more or less under control. We know that crude protein plays a role. We know how we can, let's say, reduce the problem. But suis, as in this morning was mentioned, still in many countries, amoxicillin is a standard in the diets. And we don't want antibiotics in the diets. We want antibiotics to be used only for humans and when the animals are needed. So this, let's say, approach of treating all the animals with amoxicillin to prevent suis infection, this will disappear for sure. Now is zinc oxide, and maybe next year will be this. So when we have suis infections, we have done some trials in Schothorst to understand how to avoid this situation. And I must say, when you see a piglet with these neurological symptoms, forget about the diet and treat with antibiotics. So don't try to solve these problems with the diet, because you are already, let's say, too late. So don't sell your diets in order to avoid this. No, try to sell the diets to prevent this. And if we want to prevent this, this is, let's say, a trial that we're using positive control with this amoxicillin, negative control with no additive, and then these medium chain fatty acids, let's say, combined. And then if you look to the prevalence or the animals treated with antibiotics, you see no big difference, but you see that probably the medium chain fatty acids can play a role. And as I mentioned, can play a role more as prevention when you are this situation treat with antibiotic. The most important part for Streptococcus suis is that, as you probably know, is not an intestinal problem, is more problem at the oral cavity. So the suis is already in the tonsils. So diarrhea, we always were thinking as nutritionists, okay, diarrhea is in the intestines, we need to treat the bacteria in the intestines to avoid problems. The suis is already here, and still there's a lot of debate what is the role of the intestines, but what is clear is that the suis is in the tonsils, so we need to find solutions that are really helpful already in the oral cavity. And probably, let's say, when you have these medium chain fatty acids and that are already available and they have some antibacterial effect, and you don't need to form a micelle and lipase, and this probably this can help already. Probably will not be the final solution, but at least, yeah, we know that can play an important role. The next one, please. Yes. So now we go to the conclusions of my presentation. So in fats and oils, let's say the decision of taking one or the other will be mainly driven by price, digestibility. I didn't put much, but also on the effect on the carcass. No, we see more and more that the meat is more important. So in swine we want intramuscular fat. In poultry we also want to control a bit the color of the skin and everything. So the final product will also be really important, and their fats will play, of course, an important role. The digestibility of fats is multifactorial and will be affected by the animal category. So we have seen that a piglet digests less, but in a different way than a growing pig. And also we have seen that the UR ratio is probably really important. And as nutritionists, I say, put this in your formulation program and try to be on the safe side at 2.5 at least in all the animals, and then you are there on the safe side. Formulation, let's say processing, will be more and more important, especially when we have these byproducts available. These byproducts will be more and more in the market available, so then we need to know how to use them. In the absence of antimicrobials and zinc oxide, maybe we can start working with this omega-6, omega-3 ratio to control more the anti-inflammation versus the inflammation. And this can be done mainly in piglets and lactating sows to start with. And then if you are satisfied, you can go to growing finishing pigs. The next one, please. And then when we want to put the value on the fats and oils, besides the UR ratio, it's really important the free fatty acids content. So as a rule of thumb, we say, okay, more than 60% you are in a risk situation. So then you saw in these two examples that having only free fatty acids in your diet, the fat digestibility drops. So don't do this. In the future, we will do more analysis on the fatty acid profile, total fatty acids, free fatty acids, in order to predict the right energy values, net energy or metabolizable energy for poultry. Of course, when we work with the net energy system, fats and oils have a higher value in the net energy formula, so then they are the preferred energy source, so then they will become important when we have good predictivity of the digestibility of these values. And more byproducts will be in the market. So we need to better understand the link between fiber, bacteria, dysbiosis, fat digestibility, all these kind of things. Next one. And thanks for your attention.
Thank you. Great talk, and you stayed perfectly in time. Thank you so much for that. No problem. Any questions from the audience here present? I don't see any for the moment.
So far, everything clear then?
Or everything is so clear.
Or after lunch, not much?
Nobody? Yes. In the back first, maybe, and then you can throw the microphone. That's how they call that.
Thank you for your presentation. What is your opinion when you look at the digestibility in pigs of fat sources? They are quite high, and if you have the digestibility in raw materials, they are often much lower. And you showed also some pictures, and then you could see that the digestibility in pigs, in poultry, it's lower, but in broilers. But what's your opinion about this?
No, I think if I look to the protein and starch rich, I think we feel, as nutritionists, we feel more comfortable with the energy value. With the fats, probably we need. So far, we try to understand what factors play a role. Digestibility trials, as I try to explain, are not easy because applicable. Then you will never use, let's say, a diet with 9% fat in your diet. So then you need to remain at low inclusion level, and then you have many other factors playing a role. So then probably this model approach can help us to better predict the, let's say, the energy value. And for me, what is important as nutrition is more look to the ranking. So if you say, for me, the soya is 100, look to the ranking of the different fat sources, and these at least you should be able to explain by the chain length, the saturation, these kind of things, and be really critical with the energy values, because nowadays that the feed stuff prices are high, we get often from our customers, hey, I'm offered this phospholipid mix or this, and I get this value higher than soy oil. Is it possible? Yeah, then I think probably it's not possible, and we need to better understand how to help them to say, okay, this is the level that you can use, but you have these saturations and chain lengths, and this go to that level. But let's say I think still we have work to do. For sure.
Fortunately.
Yes.
All right. There's another question here in the front.
Yeah. Should I?
You can just throw it. Yes. Wow. Great catch.
You got it. Thank you very much for your presentation. Referring to the first presentation comparing the different productions of broilers, do you have here you have focused on the different ingredients. It's very clear which one are more digestible and so on. Do you have any idea if the new broiler strains, or the slow growing, or the organic ones, they would digest the same? Do you have the same ranking, or they would have a different liver function or physiologically?
That's so from one side we say the animals need to remain longer, let's say in the farm to get the same. So in theory they should grow slower because you want a similar final body weight, but instead of 30 you want 50 days. But then that means that they will be longer in contact with the litter, and there would be more interactions with the litter and this. So yeah, we are doing trial, but of course now the genetic is just coming in the market, so the breeder quality can also be an issue. So still I think it's too early to say, okay, with the new genetics, forget over the additive because they need to grow slower and they are really relaxed and there's no challenge there, and with some, let's say, ingredients will be enough, and yeah, the FCR is less important than in the past. I think that it's too early, but yeah, this is the market where it goes. So we need to understand, what are the difference in genetics and what are the difference. Yeah, it could be some people say no, the efficiency should be less because we need to put more fiber in the diet, for example, to try to keep them long in the farm to get the target body weight and this. So there would be more interactions compared to a conventional broiler diet with only maize, soya, and fat, let's say. So now fiber comes into the broiler diets, and this would make a bit more complex. But also, luckily we have enough to do with the new genetics, and then hopefully in 5 years we understand, okay, this genetic is more sensitive for this. That genetic, better to focus on that. But I think as you bring new genetics into the market, you need some time to also stabilize the breeder farms to get good quality chicks, and now maybe you get conclusions but are more related to the breeder farms rather than the broiler itself, let's say. But it's an interesting future, let's say.
Yeah, I would like to add on that. It's funny that we call them new genetics. I have some people I would call them old genetics, but that's just a comment on the side, right? Any more questions from the audience? Yes, over there. And again, great catch.
Francesc, I had a question about fish oil. The quality of fish oil is going down.
Yes.
What's your opinion about you mentioned alternative algae oil?
Yes.
I think when is it so far that it's commercial available to use?
From what I understood, is commercial available. There's a couple of companies at least that they are promoting, and the positive thing compared to fish oil is that you should be better able to control the quality. So I think the future, like in soya, when you buy soya, you want a certain crude protein level, and you have really contracts that it says what is the quality of soya. For the fish oil, I think the future will be into that direction. If you want a premium product, put a minimum on the DHA, EPA, and they should be able with this new technology to control better the variability and to offer more constant product rather than, now you get anchovy, now the salmon is more farm salmon and the soy oil is coming, so then what you get as fish oil is not that much compared to a soy oil, and then I would say use linseed probably or another raw material to try to change the ratio a bit and don't pay too much for a product that will not bring you what you are looking for. But probably still is too early to get it in, I'm in trials we have seen it already, but maybe at commercial level still it's a bit too far or too expensive. That could be. Yeah. Yes.
Thank you.
No problem, Rob.
All right. Good question. Any other question? Yes, on the right side here. That's a sharp angle. Yeah, but still a good job.
Francesc, I have a question regarding coconut oil. You showed us a trial about coconut oil compared to soybean oil. But what is your opinion when you use coconut acid oil? Do you expect an additional effect, or what do you think?
Yeah, I think
you have some research about this.
Yeah, the main discussion is always in terms of digestibility. I think can be quite similar. Put on digestibility could be similar. Only soya oil maybe has no functional effects compared to the coconut oil, let's say. So then it's depending on the, let's say, on the booster, on the added value that you want to give on coconut oil because it has some functional effects compared to the soy oil. But in terms of energy, soya will always be higher in energy than the coconut, let's say, only depending how you want to make digestion and which matrix value you want to put, then it can be interesting or not. But in terms of energy, they are shorter, there's no glycerol, let's say, so the energy should be lower in coconut compared to the soya. But on the other hand, you have the antibacterial effects that you can give an extra value because you have antibacterial effects.
Okay, thank you.
Yes.
Can I maybe ask, it's a bit in the same direction. When I talk with production organizations, we very often have discussion how to make the decision on choosing, for instance, lauric acid derived from palm pit kernel compared to get it in from an additive.
Yes.
Do you have a formula, or do you have a way of answering that question? What is more or less, let's say, the threshold of cost of raw materials versus the additive production cost? Do you know where that threshold is? And this moment, as we have a very volatile times in terms of raw material prices, how do you see that? How do you judge on this moment the value of sourcing it from palm pit kernel compared to sourcing it from additives? Would you have an idea on that?
So we have an Excel that you can say this is the contribution of the ingredients and you get this, and then on the additive you will get how much you need to get the same. So this we have it. In the current situation, I think the prices of these medium chain fatty acids are as others, but they are sky high. So then I think if you say no, in the current situation I'm not interested too much on the energy value, I'm more interested on the antibacterial effects. Then probably an additive will be more cost effective, and then you say, okay, I use soya and other raw materials to cover the energy, and I use a solution for the antibacterial, then I think this could be more cost effective rather than including 2.5% coconut oil in your diet, for example. So this I'm totally convinced. I don't know the prices of the additive nowadays, but I'm still convinced that the coconut oil prices at the current situation are too high to replicate this trial, let's say.
Yeah. Mm-hmm. All right. Interesting. Okay. If there's no further questions, I would like to thank you again for your great presentation.
Thank you.
And I would like to ask Christy de Wildt to come on stage. And Christy will be getting us the last contribution of the day, at least a formal contribution, because after this presentation at 4, let's say starting from a bit before 4 until 6, we'll have the cocktail party here in the room adjacent to the seminar room. And then by 6, for the people here in Amsterdam, at 6 we'll have the dinner at the Jakarta Hotel. Okay, so this is last contribution here. We have time to discuss a bit with all the speakers after this session, and of course we'll have some time for questions on this presentation as well. So Christy studied animal science at Wageningen University, here in the Netherlands, of course, with a specialization in animal nutrition and metabolism. In 2020, that's not so long ago, she completed her internship at FRAmelco, and she was so happy there, and also FRAmelco was so happy from her contribution that she was able to join the R&D team as product manager for lysolecithins. Christy, the floor is yours.
Yeah, thank you. So in this final presentation, I will introduce our product Digest Health based on its composition, its benefits, and I will also show some recent trial results we have obtained. So first of all, what is Digest Health? So Digest Health is a complementary feed that consists of glycerides of butyric acid and lauric acid and some glycerol, and this is then combined with hydrolysolecithin or lysolecithins. So it is a dry product and suitable for all animal species, but for this presentation I will limit myself to only weaned piglets. So what benefits has this combination of glycerides and lysolecithins? So we all know that weaned piglets are required to grow fast and grow well in a short amount of time, and therefore energy in the form of fat and oils is added to the diet. However, in the first weeks after weaning, these piglets have an insufficient production of lipase and bile salts, and therefore cannot fully utilize this extra added energy. So this is where lysolecithins offer the solution, because lysolecithins are known to improve the nutrient digestibility by improving emulsification and enzymatic potential, and improve the nutrient absorption by improving the mixed micelle formation and the intestinal permeability. However, by improving the nutrient digestibility and absorption, you will improve the nutrient efficiency. But this only makes sense when you have a readily well-developed gut and gut health. And this is where then the glycerides come into play, because glycerides are known to affect the microbiome balance and intestinal integrity, and thereby the intestinal health. So for the first part, I will just focus on how lysolecithins will contribute to a better nutrient efficiency. But first, I will discuss fat digestion shortly, because it is important to understand where lysolecithins have their beneficial effects. So fat digestion in vivo is just a 3-step process that is mainly taking place in the small intestine, which is an aqueous environment. So it starts with the fat emulsification, during which these large fat droplets are broken down into smaller droplets by the combined action of lysolecithins and bile salts, thereby producing an oil in water emulsion. So the fat emulsification is followed by fat hydrolysis, during which the enzyme lipase hydrolyzes the triglycerides into beta-monoglycerides and free fatty acids. So the smaller the droplets are from the fat emulsification step, the more surface area there's available for lipase to act on. Also during this step is the formation of the mixed micelles. So these fat hydrolysis products are hydrophobic and cannot travel easily through this aqueous environment, and therefore they are packed together into these hydrophilic mixed micelles. And lysolecithins help here by creating smaller mixed micelles. So the final step is the fat absorption, during which these smaller mixed micelles transport the products to the intestinal wall, where they release them, after which they can be absorbed via the membrane. And here lysolecithins help by improving the permeability of the intestinal membrane, and thereby nutrient absorption. So to understand how lysolecithins will improve emulsification, it is important to understand that native lecithins, so the precursor of lysolecithins, and lysolecithins themselves are amphiphilic molecules, meaning that they have a water-loving site and a fat-loving site, of which the head groups are the water-loving or the hydrophilic sites, and the fatty acid tails are the fat-loving or the lipophilic sites. And because lysolecithins only have one fatty acid tail, they are relatively more hydrophilic than native lecithin having 2 fatty acid tails. And this has as a consequence that they differ in functionality and therefore also the type of emulsion they create. And this can be indicated with the hydrophilic-lipophilic balance, or HLB scale, and this scale only indicates whether you are dealing with a more lipophilic emulsifier that promotes a water and oil emulsion, or a more hydrophilic surfactant that will promote an oil and water emulsion. So a lower HLB value indicates a water and oil emulsifier, and the higher HLB value indicates a oil and water emulsifier. So for an optimal in vivo fat digestion, you will be needing an oil and water emulsifier because fat digestion takes place in an aqueous environment. And ideally with an HLB value of 8 to 12. So if we look at native lecithin, we see that native lecithin have an HLB value of around 4. Thereby they promote a water and oil emulsion and also fall outside this ideal range and are therefore less suitable for in vivo fat digestion. However, lysolecithins have an HLB value of 8 to 10, thereby they promote the required oil and water emulsion and fall inside this ideal range. And therefore, lysolecithins are very suitable for improving in vivo emulsification. But an improved emulsification can also be measured via the critical micelle concentration, or CMC, which is just the minimum level of surfactant that you need to add to establish an effective emulsion. So the lower the CMC, the less surfactant you need to add and the higher your efficiency is. So we determined the CMC of lysolecithins, native lecithin, and a technical emulsifier, and we found that lysolecithins have a 5.8 times lower CMC than the technical emulsifier and a 1.9 times lower CMC than native lecithin, indicating that you need to add less of lysolecithins to establish an effective emulsion. So this is just to visualize the difference between native lecithin and lysolecithins in terms of oil and water emulsions. So on the left, we added soybean oil to water, and then you see that the oil and water do not mix, so we need to add an emulsifier. So in the middle, we added native lecithin, and then you see that the oil is not properly emulsified and forms this sticky and gummy formation. Then on the right, we added lysolecithins, and then you see that this finely dispersed oil and water emulsion is created, as well as an equal distribution of the emulsified fat droplets. So lysolecithins also affect the enzymatic potential, and this is because the enzymatic potential is dependent on how well you emulsify your fat. So if you improve your emulsification by using lysolecithins, you will also improve your enzymatic potential. And this can be explained by 3 reasons. First of all, lysolecithins are relatively small molecules, meaning that they also produce small emulsified fat droplets, and thereby you increase the total surface area on which lipase can act, and thereby its enzymatic potential. Secondly, enzymes are hydrophilic and need a hydrophilic environment to function properly. So by creating this finely dispersed emulsion with these smaller fat droplets, you will also create a more hydrophilic environment, thereby again raising the enzymatic potential. And finally, fat forms also a physical barrier for enzymes involved in starch and protein digestion. So again, by creating these smaller fat droplets, this more finely dispersed emulsion, you can reduce this barrier more and more and giving these enzymes also better access to their substrates, raising their enzymatic potential as well. So to demonstrate this raised enzymatic potential, I'll show you this trial where a mixture of palm oil and lipase was used, and then either lecithin or lysolecithins were added, after which the fractions of triglycerides, monoglycerides, and free fatty acids was measured. So with the addition of lysolecithins, you see that the breakdown of triglycerides goes faster and also to a higher extent than with the native lecithin. And this is also reflected by a higher and faster release of the monoglycerides and the free fatty acids. So by adding lysolecithins, you will create a more suitable environment for, in this case, lipase to act in, and thereby raising its enzymatic potential and the efficiency at which triglycerides are broken down. So lysolecithins also improve the formation of mixed micelles. So as I said, the fat hydrolysis products are hydrophobic and cannot travel easily through this aqueous environment of the small intestine. So therefore they are put together into these hydrophilic mixed micelles, which transport the products to the intestinal wall, release them, after which they can be absorbed by the membrane. With the incorporation of lysolecithins into these mixed micelles, you create smaller mixed micelles, which are more hydrophilic and more stable than the bigger mixed micelles, and this improves the transport efficiency and thereby also the amount of products that is delivered to the intestinal wall and becomes available for absorption. So apart from working on a physical level, lysolecithins also work on a physiological level by improving the permeability. And this is because lysolecithins readily diffuse into the membrane, thereby altering the structure and functionality of the membrane. So here on the left, we have a membrane under normal conditions, so it's just consisting of a phospholipid bilayer. And you see that there's little space between the individual phospholipids because phospholipids have two fatty acid tails. And this increases the viscosity of the membrane and decreases the permeability and thereby the nutrient absorption. So when we incorporate lysolecithins into this membrane, you can see that there's more space created because lysolecithins have only one fatty acid tail. Thereby they reduce the viscosity of the membrane and improve the permeability and the nutrient absorption. Secondly, lysolecithins have a conical shape as compared to the cylindrical shape of phospholipids. And this results in more bending of the membrane, and this is important because the bending of the membrane makes it easier for the membrane to form new protein channels that can be used then for atom and nutrient absorption. So by creating more space, more bending and promoting the formation of new protein channels, lysolecithins will improve the permeability and the nutrient absorption. So having discussed now how lysolecithins benefit nutrient efficiency, we will continue with how the glycerides then will do their part on intestinal health. So intestinal health and the intestinal integrity are, of course, key to improve the profitability. So logically speaking, when you have a well-performing gut, you will also maximize your production output. However, bacteria and the toxins they produce can cause severe inflammation, break down the tight junctions between the enterocytes of the intestinal epithelium, and thereby causing a breach in the barrier against the external environment. This has as a consequence that bacteria and toxins and undesired proteins can pass from the lumen into the circulation and, of course, affect the intestinal health and the health status of the animal in general. Luckily, we have glycerides because glycerides of specifically butyric acid have the functionality that they support the production of tight junctions, thereby improving the barrier and preventing these pathogens and other harmful factors from crossing into the circulation, thereby supporting a healthy gut. Furthermore, these butyric acid glycerides also stimulate intestinal development by increasing the villi height and improving the villi height to crypt-depth ratio. Thereby, you will also improve the nutrient absorption. But these changes also are associated with a more healthy gut. And then finally, glycerides are known to affect the microbiota composition, so where the antibiotic growth promoters will completely destroy the microbiota, the glycerides are more selective, so they will stimulate the growth of the beneficial bacteria while inhibiting the growth of the pathogenic bacteria. And of course, a healthy ratio between beneficial and pathogenic bacteria will also stand for a well-balanced microbiome. And the well-balanced microbiome will be able to activate and stimulate the immune system, offer protection against these pathogenic bacteria, and also offer some energy and nutritional support to the animal. So now I've discussed the mode of action and the background of this product, and now we'll just show 2 trials in weaned piglets with this product. So in this trial, we did this at a commercial farm in Denmark. We had 340 weaned piglets that were weaned at an age of 26 to 28 days, and they had an average start weight of 7.8 kilograms. So we had 2 feeding phases, a starter phase for the first 2 weeks and the grower phase for the final 4 weeks. These piglets were equally divided over 2 treatment groups, a control group that received the basal diet and the treatment group that received the basal diet with 3 kilograms of the product in the starter and 2 kilograms of product in the grower period. And also no additional health-stimulating factors were added to see the effect. Yeah. So if we see, if we look at the results, we see that the piglets that received Digest Health had a higher final weight with an increase of 5.9%, and this was of course associated with the higher average daily weight gain of almost 8%. And this higher growth performance can partially be explained by the higher feed intake of almost 5%, but also the piglets utilized the feed more efficiently by, as indicated by the significant reduction in FCR of almost 5 points. In the second trial we did at Insolpig in Belgium, here we had 512 weaned piglets that were weaned at 21 days of age, and they had a start weight of 5.5 kilograms, so these were relatively small piglets. Again, we had 2 feeding phases, a starter phase the first 2 weeks and a grower phase for the final 5 weeks. And again, the piglets were equally distributed over 2 treatment groups, with the control group was given the basal diet, and the treatment group got a basal diet with 2 kilograms of Digest Health on top. And again, no extra health-stimulating factors were added. So again, we saw that the Digest Health piglets had a higher final weight with an increase of 2.2%. Again, a higher overall average daily weight gain of 3% increase. And this time it was not due to the feed intake, as there was relatively no difference between the 2 groups. But again, the piglets were more efficiently using the feed with a reduction of more than 5 points in FCR. So in conclusion, we can say that Digest Health can help alleviating the weaning stress of the smaller piglets, for instance, by improving the microbial control and intestinal development, thereby giving the lysolecithins the opportunity to fully improve the fat digestion and nutrient absorption better, and let the piglets grow towards their genetic potential. So this was it. So thank you.
Thank you so much. For sure, you stayed in time, so that gives a lot of room for questions. Who will kick off? Yes, on the right side there.
Thank you. Just a short question on the combination of the lysolecithins with the glycerides, because you say that the lysolecithins enhance the membrane permeability, but then you add the monoglycerides to increase the tight junctions. So it seems a bit counterproductive.
Yeah, the lysolecithins will not affect the tight junctions, so that's really for the glycerides to improve the intestinal integrity. That prevents then the pathogens from entering into the bloodstream. And with lysolecithins just improving the diffusion of the nutrients over the membrane. I don't know, is that answering your question, or is it more like, will the pathogens then also cross the membrane?
Yeah, that's what I was going for.
For now, we have no indications that this is indeed the case. We have not seen it in our trials, and we have not gotten any feedback on it. And also, based on the paper that was done in rats that added lysolecithins at higher levels to study how the pathogens will react on the membrane, we have to increase our dosage level like 6 to 12 times. So we don't suspect that will ever happen.
Okay, so in conclusion, you would say that the increased membrane permeability will not lead to any more toxins or more maybe leakage in the intestinal tract?
No, and it's of course key that you have a good quality feed, of course. Yeah.
Okay, thank you.
All right. Next question. Yes, here that's on the right side.
Thank you. I have one question just to connect it with the presentation of the speaker before you. Mm-hmm. He mentioned that when there is a lower intestinal health, that the bile salts will be lower. How do you see that this product can help to solve this problem?
Well, by adding lysolecithins, of course, you add more emulsifiers, so you will be able to opvangen.
To
cover some of the reduction in bile salts, and of course, with the lower intestinal health, you have the glycerides that can push for a better health status, and then you will hopefully get the bile salts back on track.
Okay. Thank you.
Okay. Other questions? Yes. This side.
Can you catch?
Throw the micro. Yes. Great.
I had no question about the experiments.
Mm-hmm.
What was the mortality? Was it equal or?
Yeah, there was no difference between the treatments. No.
And when there are health problems.
No, there were no severe.
But if there were health problems, what could this product contribute? When there are really big digestive problems.
Yeah, I was going to ask something similar.
Because when there are not problems, you can test, but often you see no difference.
Yeah, these piglets were not challenged or anything.
And would you expect, let's say, a higher return on investment when your pigs get challenged in with gut health, or how would you judge that? I was going to ask something in the same direction.
Yeah, when they are challenged, of course, then this product would be a solution, of course, and then I expect a higher return on investment, of course.
Mm-hmm.
No.
But you haven't done experiments yet.
We haven't done experiments in that.
So
No, that's just
promising
speculation.
to maybe set up a trial where you have like combination with different types of challenge.
Yeah, that would be interesting. Yes.
All right. Okay. Other questions? On the back there. Okay.
Yeah, thank you for your presentation. I have a short question. You showed the scale of the HLB values, and the lysolecithins lies on the right side. But I was wondering if they have a certain specific range or one value for the lysolecithins, or and also which value would you say is best for digestion for the oil in water emulsification?
Yeah, so optimal for oil and water emulsification is 8 to 12. Mm-hmm. So inside that range, yeah, I haven't seen any differences yet, but maybe there are, but I can't say for sure, but that's the optimal range. So if it's lying between those values, then it's fine.
But for lysolecithins, do they have a value or a range?
Yeah, lysolecithins lie between 8 and 10 depending on the type of lysolecithin, but they are always 8 to 10. Yeah.
Oh, okay.
Perfect. Any more questions? Yes. On the left side.
Thank you, Christy, for a nice presentation. You were explaining a combination of lysolecithins and glycerides. You were explaining now about piglets, but what do you see if potential for this product for other species like broilers? Or
Yeah, we have yet to do some research in broilers as well, but as we have seen that our glyceride products also work in broilers and our lysolecithins also work in broilers, so the combination will also probably work, but we have still to generate some values for that.
And do you also see if we want to replace zinc oxide, which is a big item at the moment for piglets, that this product should be used when you are replacing zinc oxide in piglet diets?
Yeah, it could be a possibility.
Okay. So I have the impression we have a lot of work ahead of us.
Yes.
So, you just got started, so a lot of work in front of you. Any more questions for the moment? No? Then I would like to invite all of you here in the room to join us in the Ghent room, where you can go further into discussion and also entertain the different speakers of today, the excellent speakers of today, with more questions and more discussion. And with that, I would like to thank you all, of course, being here present live. I hope to see you also next coming days during VIV, I think most of you. And also the people online, we would like to thank you a lot for your presence today. And we hope you have enjoyed this very informative session. Thank you so much.